clinically. And there comes the research. Given that the trials are very mandatory, but 80% of the trials are being conducted in only 20% of the research institutes. Nevertheless, many investigators who have that enthusiasm or time are left behind. So we have developed a platform called as Ignition, which is going to help you there to ease your time, and whatever time that you have, the smallest time that you can spend for research can be used judiciously. So in a conventional method of doing a trial, a lot of activities, it's a huge sequence of activities which starts from the protocol designing, site selection, the laboratory selections, finalizations, regulatory approvals, including the DCGIs, as well as the institutional ethics committees. Then the actual patient enrollments also happens. So that's where your important piece of contribution comes into picture. So based on the protocol, the inclusion and exclusion criteria of the trial, you are going to select the patients who are eligible for the trial, and then the actual problem of the documentation happens, which is a huge, which most of you are aware that a clinical research has a lot of documentation. That's where you cannot spend much time. There, the TriMix platform is going to help you a lot by easing those documentation procedures, digitalizing the entire process. The next steps of that is data cleaning and data validation of the entire patient enrollments or whatever happened till then, the data log, and then the analysis, the statistical analysis. Important part of this is the research publications. So once there are many pros to do these clinical research, just not for, because I know most of you are not looking at the financial aspects, but the paper publications post the completion of the research or the trial. So there comes again the TriMix part, where we are going to publish the papers of the trials conducted and also help you to have a better visibility in most of the journals that are of high repute. That also helps you for the global networking and also associations like American Diabetes Association and others, because you're already visible across through the trials that you have conducted and also through the research publications. The problems with the clinical research and the essential steps that are involved there have a lot of duplication of the work, where the research coordinators who are your team members or yourself is doing that activity, you may feel that why am I entering this data again and again? So at the same, because as you know, the research has a lot of documentation and endless paperwork. Important thing is about your participation or your contribution in the patient enrollment. The gaps there are so critical that it will hamper, and that's where the TriMix platform is going to help you to identify the patients through the electronic health records that a platform that we are going to provide you, so that there is an easy flow of all the patient database, and you can filter those patients who are actually eligible into the trial. So that is going to help you, which is not happening in the conventional clinical trials. That is one of the plugs or one of the bottlenecks that we have identified, and we are trying to fix that. And we have successfully done that through 3 clinical trials so far. And also, there are a few other items. For example, the investigators being burdened with, like 80% of the trials in India are being conducted only in around 20% of the institutes. So that is like same investigators are doing the trials again and again, but there are a lot of enthusiasts like you who are interested but not sure what are the next steps to do that. So we are here to help you hand in hand, help you with the next steps of the first steps of doing a trial, because we have a few sponsors who are ready to do the trials in India. Our sponsors are US based, globally from Australia as well. And so far, we have recently concluded a trial, which I'm going to explain what about this trial in the next slide. So that's where Triamix has identified those problems, and we are a startup company started off in 2020 by a couple of IIT Roorkee graduates and also a medical oncologist, backed by US-based investors. We have around 20 to 25 of software engineers coming from IITs who have developed a software platform that would definitely help you to shorten the time, or whatever time that you could spend can be judiciously used for the clinical research purpose. And also we have the platform which is also interfacing with the ethics committees, the patient pools, the lab, the central labs, wherein you need not as such, or your team need not transcribe each and every value of the lab parameters manually. The software is so integrated that the lab values will automatically flow into the database, so that there are no transcriptional errors, no manual entry errors. The quality of the data is very high, and the sponsor is very happy to see such kind of a good clinical research being conducted that will help their product to be marketed or launched without any hurdles from the regulatory inspectors, who are going to ultimately the decision makers to allow that particular drug into the market. And I'm going to give the next slide to my colleague, Mr. Vikram Bhatt, who is going to explain how we are going to bring that value addition or make that change and fix those bottlenecks that we have identified earlier. Over to you, Mr. Vikram.
Thank you, Dr. Suresh. I'm Vikram. Thank you for elucidating all the facts about what are the issues that are faced in clinical trials. So right now, I'm going to tell you how Triamix technology is going to help in making sure that these issues are not there. So initially, I would like to start with what a super site is. A super site is a clinical research ready site. A super site can be a clinic, a multi-speciality hospital, it can be a medical college hospital as well. So at these super sites, we've made a bunch of tech interventions and eased the flow for quick conduct of trials. Super sites also network with one another to deliver synergies. Like-minded investigators can collaborate with their peers, both nationally and internationally. Investigators conduct similar type of research in the same therapeutic area and share their know-how and brainstorm effectively. Due to this, the investigators receive new info related to the field of their expertise, newest developments in the upcoming therapies, gain access to new drugs and therapies, and become part of associations like the American Diabetes Association, the Indian Diabetes Association, etc. By becoming a super site, PIs are able to leverage the power of these associations. Overall access to these networks helps the investigator build a very robust clinical research profile and gain visibility amongst industry sponsors. PIs can gain access to company-sponsored studies and also be published in national and international journals like Nature, Science Direct, etc. One such advancement is the extensive use of CGM and wearables to effectively monitor glucose levels and metabolic health in treatment of diabetes. Dr. Suresh will explain further with regards to that. Now, I'm going to explain how super sites, or rather how Triamix helps its own super sites. We have a three-pronged approach. One is the network, the other is the infrastructure that we create, and third is the support system that we provide. I'm going to talk about the network. So initially, we spoke about a different type of network. Now we're going to talk about the peer-to-peer network. We have pre-built a network of independent consultants, SMEs, key opinion leaders, peer network, and regulatory experts. These networks help investigators to get the necessary approvals in an instance. Our platform has made research proposal possible in one click. Hence, there is no harsh task of printing, signing, sending the dossier here and there. It can all happen in one click. We've tied up with multiple independent ethics committees as well. Now, a lot of investigators here would not have clinics. And the question is, if I do not have an ethics committee in my clinic or my hospital, what do I do? We help you tie up with independent ethics committees, and we can conduct clinical research there as well. A bunch of hospitals which already have an institutional ethics committee. Our platform can coordinate with those guys as well. Query resolution can also be done on one single click. Now, one more issue that happens is with respect to patient identification. Now, considering the inclusion and exclusion criteria is very robust, it is very difficult for investigators to find the right kind of patients who fit into the inclusion-exclusion criteria. So, Triomics has a one-click query search. So, you just need to search there, and you would be able to get a list of your patients through your EMR. And these patients are pre-qualified to be able to be recruited into the trial. Triomics, we've digitized the inclusion-exclusion criteria, making it easy for you to recruit patients as well. Then comes the patient management. Once these patients are recruited and they've signed their ICF and they're a part of the trial, managing these patients is also a tedious task, considering you're also doing your clinical practice. Patient journey during the trial can be easily managed. Complete overview is available to the investigator, including visits, scheduled visits, tests, and investigations too. The third thing, what we've done is we've digitized the CRF. Most of you would have, whoever has done trials, has used a paper form of CRF where the investigator writes something into the paper CRF, after which it is a third-party person, will have to key in whatever these details are into the computer, followed by which a monitor comes and says whether whatever has been keyed in is correct or not. This we call as monitoring. This is time-taking, very tedious, and if there is any issue with the monitor, the investigator has to be there to understand what it is and what it is not. So what happens with Triomics is we have done source data verification. Since we've digitized the CRF, source data verification has already happened, because in the CRF, the investigator itself enters whatever details are there. We also have tied up with regional and local labs. So if in case there is some lab tests or some diagnostic tests that need to be ordered, this can be done on one click through the tab. So there is information which is sent to the local lab saying that this test needs to be done, and the person from the local lab comes and performs the diagnostic test wherever it is, and the details are auto-populated into the CRF. We also have a very good support system as well to conduct the trial. We have experienced project management team which will handhold you through the whole duration of the trial. We have a quality assurance team which does the monitoring and the audits after the trial is done. If in case you are interested in investigator-initiated trials or IITs or academic trials, we can customize the CRF for you. That also is done. We can provide you 24/7 support in terms of any questions that are there. We'd be more than happy to answer. And we also have one coordinator who is trained with Triomics technology at your site. We also have a statistician which will be provided. So these are a bunch of benefits to becoming a super site. You gain access to global clinical studies. You would become a prime choice for industry-sponsored trials. You would earn honorarium in conducting these trials. We help you with our EDC, which is our software for conducting these trials. And we also help you with patient identification and patient management. Over to you, Suresh.
Thanks, Bhatt. Thanks that's so much for giving a big insights about the super site. We have next few testimonials, a few investigators who have actually done trials for us across India, and a couple of their experiences doing trials or research with the Triamix is for you here. Meanwhile, we have some technical, but meanwhile, I'll call upon one of the investigators we have here, Dr. Srihari Kulkarni. He has participated in a couple of trials for us. Both of them, one is sponsored by an Australian sponsor and another one by an Indian sponsor. And he has used our platform and he will share his experiences.
Thank you, Suresh. Good afternoon, everyone. I'm Dr. Srihari. I'm a practicing diabetologist in Bangalore. Clinical trials we've been doing since ages, and we know how cumbersome clinical trial was earlier. 5 years back when I started my clinical trial journey, the main area of problem was the storing of the CRF so that it does not get wet or all other issues. But now with the new onset of Triomix, the investigation is digitalized. I have done two studies with them. One is RESAP study where it's done for the COVID, and another is a ultra-human study. With both of them, it is very easy. You don't have to do anything, and all data is stored in a digital way. And one good thing, one more additional is they provide a on-site CRC. If you don't have a full-time CRC or you don't have CRC, that is one advantage. Triomix gives the CRC, she is there full-time, and I have interacted with 3 or 4 CRCs. I can bet you they are good. All CRCs, they work very well, and one of the CRC is very advantageous what Triomix has done. In one RESAP study, I conducted in 1 month, had 200 patients. We could finish it. I still remember when Dr. Suresh called me one night and said, "Sir, we need to do this study." And I said, "Okay, when I'll start." But he said, "Sir, we need to finish it in 1 month." And first we had to have 100 patients, but we could go on up to 200. So easy it is. And the CRC were very cooperative. I can say that they were there from morning 7 o'clock till night 11 o'clock also till we completed the study. Last but not the least, remuneration part. I don't want to reveal, but it's very good, and it's on time or even before. So with this, I think Triomix, wish you all the luck for your future endeavor. Thank you very much.
Thank you, Dr. Srihari. Thank you so much for those words. We have few other investigators who couldn't make it today. Yes, please. Please talk.
Like NHS, NHS has, I don't need a speaker. NHS has the uniform EMR record. In India, we don't have. Every company has a different EMR record system.
Correct.
And to collect data and process where you have a free methods of collecting data into your system, will need a lot of setup.
Correct.
Data mining and putting it done.
Correct.
So how do you take care of those parts?
Definitely.
And secondly, when you say that you do secret verification from the records of the hospital, operating records or the lab records, how do you take care of the
Got. That's a very, very good and valid questions, definitely. We are going to address that. We have a couple of slides left over. We'll finish that and then going to answer those questions. Thank you. So there are other two investigators from Chennai who couldn't make it today here, but they have participated in our trials and they have something to say for you.
Hi, this is Dr. R.P. Rajesh, consultant diabetologist from Ardhavan Diabetes and Research Center, Chennai. As of now, I have conducted 2 clinical trials with Triomix. Initially, I was very much inclined to do a clinical trial, but unfortunately, because of lack of time, I had a very hectic clinical practice, and because of that, I was not able to find good time to do clinical trials. That was the time Triomix actually contacted me, and after that, I started doing clinical trials with the Triomix. Then everything became very, very simple. They saved a lot of time for me. Triomix saved a lot of time for me, and now I am able to efficiently conduct a clinical trial along with my clinical practice. And one more thing is that they have an excellent coordination with the central laboratory. So each and every time we need not call the central lab to collect the samples as well as to receive the reports. Everything was easily done just with a single click. And another important highlight is that most of these works are paperless. So we don't actually have to find a storage space to keep all the files in the clinic. So most of these things are paperless. And another thing is that whenever we do a clinical trial, the monitoring takes a lot of time. It consumes a lot of time. So what happened with the Triomix is that most of the monitoring was actually done remotely, which saved a lot of time for us. So considering all these advantages, I am eagerly looking forward to do more and more clinical trials with Triomix in future. Thank you.
I'm Dr. K. Bharanidharan, Senior Consultant Diabetologist, Supra Diabetes Care and Research Center, Chennai, and also I am consultant in Kaveri Hospital, Chennai. I have conducted one clinical trial in Triomix. The concept of super site from Triomix is a plug and play concept, which is quite interesting. Everything happened digitally. All the reports has been sent digitally, which I can just view it in my tab, and it's hassle-free. And I can also access all the records, which really helped me in doing a research in the clinic, which I just appreciate from Triomix.
To answer, the session is open for the question and answers, and to start with the question raised by Mr. from the WOCAT, I will invite my colleague Aniket Jaiswal, who is going to answer your question, sir.
Thanks, Suresh. Good evening, afternoon. So basically, you have 2 questions. One was on the data transformation, which is basically, I can say standardization of the data across different hospitals, and one was like the identification of the patient and their mining operations. So basically, as part of our super site process, we do 2 levels of transformation. So when we convert any hospital into super site, we do a mapping of whatever standards they are using into our standard coding. So let's say one hospital is using a Dolo for paracetamol, one hospital is using Crocin for paracetamol, or one hospital is using C as a paracetamol. So we map all the 2, 3 or whatever different terminologies they are using into a standard coding, which we have mapped as part of our process. So these standard codings are LOINC, ICD, SNOMED, CT, or other standards which are used at a global scale. So whatever hospital are using, irrespective of what, when we have the mapping at the central level, so let's say Dr. Srihari or Dr. Barney or Dr. Rajesh, for a same parameter, they are using different terminologies. So they will interact with their own terminology, but at our end, we will receive in the standard format, which we have defined. So this level of mapping we do at the time of the onboarding of the super site. That is the process of the transformation. So it takes generally 15 to 20 days because we have developed our own interface engine, which makes the entire process of transformation from one terminology to another terminology very efficiently. So hope this answers your first question. In terms of the second question you have asked, like how we are handling the identifiers, because patient safety and patient confidentiality is the paramount process in any clinical trial, and that is something which is a challenge across all the clinical trial practices, and due to which majority of the sponsor or majority of the pharma company don't want to digitize the processes. So what we have done is, like as Suresh has mentioned, we have a very great engineering team. People are from IITs and BITs who have a very great engineering mind. So we have devised the platform in a way. We do segregation of the identifier and non-identifier data, and we only receive the data which is non-identifier from the hospital. So as part of removing the PIIs from all the databases, we do that level of segregation whenever we are having any query. So we are using AWS technology for those fronts. So it's a very standard process which happens in US market and a UK market. So same level of standardization and transformation we are implementing into Indian market. Happy to answer any other questions. There's some questions.
I think more of a comment. Thanks for being a part of KRSSDI. I think we always have inertia. We always believe in just practice, practice, and practice. And we, as one of the other doctors who was testimonial, he was telling that we are a very busy practitioner. I think by doing that, we are losing sight of many, many things in medicine itself. Forget about in life, we have lost a lot of things anyway, thinking of practice. So I think that is where we need to be careful and start having inclination towards practice. I was fortunate to work my initial, just after passing out, with good clinical practitioners and clinical researchers like Dr. Arpandev. And so I've been there in touch with clinical research from almost 20 years since the time I graduated. So, and then initially then a lot of DCGI rules and regulations made it almost impossible to conduct clinical research. And now I actually conduct research only in Manipal Hospital.
Okay.
So there are, these are the one things which we need to first open ourselves before we embark into the details of clinical research. That's one thing. And in the coming days, we will do one platform to run, that's in my mind when we, through KRSSDI, that how do we include many other things in clinical practice. Clinical research, it has to be managed. Even we were not digitized, I and Arvind are working. So initially we thought it's a waste of time. I also used to think digitization is a waste of time. But when we start doing it more often, it actually saves time. And of course, our handwriting is not the best in the world. And so even on that legally, and when you have the data, only you can act on it further. So I think digitalization should be the forefront of all our practices first. I don't know, many times we struggle to read what we have written sometime back also. So then comes the clinical research, CRCs, and then it was so much of a mandate few years back, especially. It became too bad that now I do clinical trials just to keep afloated, be in touch with clinical trials than for the monetary gains. Even that way, it is not so attractive for us to get into that. And I have had some interactions with him, and I think it's a very good platform. A lot of people are here whom we can share the data, and then we'll see how we can take it forward. Thank you.
Thanks a lot. Thanks a lot, Doctor, for kind words.
So doing clinical trials is one aspect. Like what about the publications?
So, yeah, so that's a very interesting question. So basically, as part of our session also, what Vikram has explained that we have, as part of our ecosystem, we have a network of medical experts and regulatory experts who help you to transform your SDTM data sets or clinical data sets into standard formats, which we can use to publish as part of standard journals or international journals. So our medical and regulatory teams help you to basically find out which journals will be the suitable for your specific research and which type of publication we can target in terms of the novelty of the research or in terms of the novelty of the clinical practices. So that is something which our medical and scientific team are helping you on day and night.
And also the topics, in the sense we as a doctor, we tell you these are the burning issues and all those things. Do you have supports, like do you go through the journals, which is the topic which can be done, the particular study, this is attractive, this can be publishable, this is replicable. Main thing is, publishing is not a big thing nowadays. The publishing data has helped anybody in clinical practice, that is all.
Exactly. So basically, finding out the novelty in the process, like whether this particular same research has been exactly done or not. So as part of our protocol development process, our medical team do a very robust research in terms of whatever existing research has been already done similar to that process. One of the study which Suresh will explain, where we have done study with CGM on healthy and pre-diabetic population. We have faced the similar type of issue because a lot of research has been already done in US and European market. But we have found out some novelties, like what additional biomarkers like cortisol, HOMA-IR, lipid profile, we can make a correlation with the CGM data so that we can create an entire metabolic profile of a patient, which help you to identify whether what is the percentage of chances of you being from diabetic, pre-diabetic to diabetic and healthy to pre-diabetic. So that level of novelty we have gained through that extensive research, and now we are in process of to publish that similar research into AD and Nature also. Abstract submission has been done, so we are just waiting for the final confirmation.
Yeah, because that is the future, because publication is one thing. Future people are asking like, okay, you have published this particular data. How this published data has helped the clinician or they help the patient in their day-to-day thing. Good concept.
Exact. And also one thing is around the data quality also, because a lot of time the international journal don't have the trust on the data which has generated at clinics or local labs. So what we have done is, as part of our network, we have integrated with CAP aggregated labs. So you need not to perform clinical trial specific or clinical research specific test at local labs or just NABL labs. Using our platform, you can create orders and you can, so they will come and they will receive the sample and they will perform the analysis, and all the reports will be automatically populated into the platform itself. So you need not to get the report, manually enter all the parameter into the system. And the other advantage which we are leveraging using this type of infrastructure is like very robust mechanism of screening, because a lot of time if we miss one parameter, let's say if inclusion exclusion criteria is on the fasting blood sugar, HbA1c, OGTT or hemoglobin. So if there is a disparity in the data that the lower limit was 12, but you have mentioned 12.1, but in the actual report it was 11.9. So the patient is eligible, but actually the patient is not eligible. You perform the study with the patient, but when the sponsor re-verify the data, the patient is not actually eligible. So using this type of infrastructure, we are making the entire process 100% leakage proof, because all the data is directly coming from the verified system. And the tunnel which we are using to transfer data from central labs or regional labs to our system, it is validated and verified by 21 CFR Part 11 systems and FDA guidelines. So this makes the entire process at a global standard, like we can use that data for FDA submission or global submissions.
At the outset, I'd like to thank Dr.
GCP certification, not just in paper, but in practice. And few months back, I had got mine. So if you're in active research, you got to change my mother. You can transfer that to them. That will be easier. I don't have a you have? You can use this. It will not allow. So you have an attachment?
No. What I will do, mail it. Whom do I mail it? doing clinical studies. My thought is that if we have the right intention and the right resources, clinical trials are never difficult. They are as easy as you want to do, but you should have a proper training of the protocol, what you are doing, and you should have a proper knowledge of GCP. Then your study becomes very, very easy, and you can conclude a study without any problem what you have. Is it okay? No, I don't want only presentation. It is coming. I don't understand your language. Can I mail it to you? Can I mail it to you? Mail, email? Yeah. Huh? What's your email ID?
Skyline.
Huh?
SKYLINE.
Skyline. Huh?
AV
AV
at gmail.com.
at gmail.com.
Type C. Sorry, correct. Charge chala.
Sorry for this glitch. कोकण देना. Thank you. So good afternoon all. We had a very interesting session where we discussed about how data is being transferred from the source data into ECRF form. EDC has been functioning for quite a while now, and the challenges what we had in India is about the same, about transferring the data from source data into a format which is being data mined and being transferred into a system, and we have a proper flow of the data, because setting up the entire system takes time and there's a cost as well. But I'm sure that we are all moving towards digitalization of data, and that's how we're going to move in as well. So I will not go into my CV. So I have done more than 110 studies. I've got 16 years of experience in clinical operations. I head the clinical operations, regulatory, PV, and QA of OCAD. I'm also MBA graduate. I've also completed my master's in artificial intelligence. Currently, I'm pursuing my certificate from strategic management, and I'm also doing a PhD in management as well. So that's my background. So GCP is actually takes about a couple of days workshop, but we are trying to finish in the next 20 minutes. So I'll take you to the definition of GCP. So what I've done is I've broken just GCP definition into parts, where I say it's a standard for designing the protocol, for conducting the study, for performing the study, for monitoring of the study, and auditing and recording and analysis of the data. So these are very, very important part when we say what is GCP. Anybody can tell me any idea what is the difference between audit and inspection? Anybody? So audit is or monitoring is a part which is done by the sponsors. A sponsor, CRA, when they come to your site, they do monitoring. When the QA person comes to the site, your site, to do the data verification, they are called as auditing. Whenever a FDA official comes, whenever from a state FDA or from a regulatory authority or from US FDA will come to your doorstep for looking to your data, that is inspection. So as an investigator, it is your responsibility to allow all the monitors or auditors or inspectors to come to your site and evaluate the data. So we have to report clinical trial that provides assurance that the data are credible, accurate, and the rights, integrity, and confidentiality of trial subjects are always protected. So this is a definition of GCP. We follow ICH GCP E6 R2 guidelines. It is based on Declaration of Helsinki and assures protection of human subjects. So we also have international harmonization aimed at ensuring good quality, development of safe and effective medicines, registration in most efficient and cost-effective manner. We all went through COVID a couple of years back, and we also had a vaccine which came up. But do we know that the Oxford vaccine, the study was done in UK. But because India follows International Conference on Harmonization, ICH GCP, even they did a phase 3 study, we did not have to do a study from phase 1 to phase 2, phase 3, or a preclinical. We directly did a bridging study in India, and the vaccine was approved in India as well. Because of the racial system, we at least had to do a bridging study. We did not have to do an entire gamut of clinical trial from preclinical to phase 1, phase 2, phase 3, and the product was marketed because India is part of the ICH GCP guidelines. So what is GCP? It protects trial subjects or patients, credibility and reliability of studied data. So when I'm saying subjects or patients, subjects as in when we are doing a phase 1 study on human volunteer, we call them as subjects. Whenever we are doing post-marketing studies or observation studies, we call them as patients. So there's a difference between subjects and patients when we are reading a protocol. So as an investigator, we should understand that what is a term being used by a sponsor or a medical monitor when he comes to your site saying that, doctor, I want to do a study with healthy subjects or patients. So we need to understand that whether he wants to do a phase 1 or a phase 2, phase 3 study, or he wants to do a phase 4 or PMS kind of study. Okay. What also is important is that whenever you follow ICH GCP, it ensures that the data is credible, reliable, and the study data can be published. So when we had this question in the last session as well, that how do we have a good publication? So whenever we want to do a good publication, first is that we should have a good protocol in hand, a good properly scientific design protocol. Always if it's an interventional study, a randomized study, the chances are good that your study will be published in a good journal. But if you follow ICH GCP guidelines, you have all the regulatory approvals, you have all the ethical committee approvals, then of course your study will be published in a very good journal. Okay. So why GCP? It protects the right and safety of subjects. Trials are based in good science and well designed and properly analyzed. Trial procedures are properly undertaken and documented. Assures integrity of data submitted to regulatory authorities. Presents a record of the trial, that is source document. What if GCP is not followed? Subjects who can take part may be at risk. You may have a wrong study which is approved or wrong medication which is approved. Failure to obtain approval of useful drugs. Suppose you are not doing ICH GCP properly. There could be a chance that the drug is very good, but you did not get approval because you do not follow ICH GCP guidelines. Costly litigation if patients are harmed, especially in US, if you do not follow ICH GCP guidelines, doctors are also barred from doing clinical trials and also clinical practice as well. So who are the stakeholders? The stakeholders are the investigators, investigator team, the regulatory authorities, CRO, the IRB or IAC and the pharmacist as well. You would imagine why a pharmacist, because a pharmacist is a person who stores the drug at your site. Suppose it's a blinded study, it's a randomized study, even the pharmacist has to follow the ICH GCP guidelines and ensure that he follows the protocol and there is no deviation in terms of storage of drug, in terms of dispensing of drug to the patient as well. So even the pharmacist also is a part of ICH GCP to be followed. Principles of ICH GCP. Clinical trials should be conducted in accordance with ethical principles of Declaration of Helsinki. Before a trial is initiated, foreseeable risk and inconvenience should be weighed against the anticipated benefit. You may have a very nice protocol, but you have to ensure that the benefit is more than the risk involved. So all those has to be taken care whenever a protocol comes to you. So whenever a CRO or a sponsor comes to you with a protocol saying that, doctor, I have a protocol to undergo, you as an investigator best judge to tell him that, no, this protocol has this lacunae, or you have to maybe change few inclusion criteria or few exclusion criteria or few study design so that the risks are less. I'll give you an example. Suppose a patient who is undergoing chemotherapy and you say that my drug wants a patient to come to the hospital on a weekly basis. Is it really justified? Because chemotherapies are given on maybe after 3 or 4 weeks when a patient comes to the hospital. If you call a patient who is undergoing chemotherapy, who's undergoing oncology treatment on a weekly basis to a hospital, that's a wrong protocol which we have designed. So we have to, as an investigator, you have to tell the sponsors that, sorry, the protocol is not right, you may have to change this part of the protocol. So always the investigator or the medical monitor is responsible for the design of the protocol which is being undertaken. The most important part whenever a clinical trial is done, rights, safety and well-being of the trial subjects. Whatever trial you do, you may be spending billions of rupees in doing a clinical trial, but if the drug is not safe, you cannot go ahead with the study. I was doing a study when I was in Merck with a phase 3 study on multiple sclerosis. It was a very good drug. But it had certain safety issues. We from the company as a sponsor had to stop the study after phase 3. We had spent hundreds of billions of rupees on this drug because it was an international trial globally conducted, but we had to stop the study as well. We also should always have the available non-clinical and clinical data. Suppose some sponsor comes to you or CR comes to you, sir, saying that we have a phase 3 study to be done. The investigator is always liable to ask the sponsor or a CRO that please provide me the phase 2 data, please provide me the phase 1 data, please provide me the preclinical data. After going through all the data, then only you should be able to start the next phase of study as well. Clinical trial should be scientifically sound and described in a clear, detailed protocol. So always you should say that the protocol what you have is a detailed protocol which tells you the entire aspect of the protocol, the medicines, the IPs, the storage conditions, the visit schedules, etc., etc. A trial should be conducted in compliance with the protocol that has received IRB or IAC approval. So no trial, be it IA study or intervention study or a phase study, we cannot start a study without a IRB or IAC approval, right? Regulatory approval at times is not required. As I said earlier, for IA studies, regulatory approvals are not required, but for any study you do, you have to take an IRB or IAC approval. Anybody, what is the difference between IRB and IAC? So IRB is the ethics committee which is associated with the hospital. Or ethics committee is not associated with any hospital. Independent working is IEC, Independent Ethics Committee. The medical care or a medical decision made on behalf of subject should always be responsibility of a qualified physician. The protocol may say that this is a rescue medication, but as an investigator, it's you who will judge and decide what is the rescue medication given to the patient, what is the care which has to be given to the patient. Each individual involved in conducting a trial should be qualified by education, training, and experience to perform his or her respective task. So every investigator who is doing a study has to be GCP trained before he initiates a study. Freely given informed consent form. We always have to seek an informed consent from the patient, and it will be a freely given consent. We cannot coerce or push the patient to give us an informed consent. We have to always explain the nitty-gritties of the protocol and take the consent from the patient. All clinical trial information should be recorded, handled in a store that allows it accurate reporting, interpretation, and verification. Verification is very, very important. As I said earlier, inspections monitoring and audits can happen outside. So you should allow verification of the data as well. Confidentiality of records should be always maintained, and the privacy and confidentiality of rules of operation should be always taken care of. IP, the investigation product should be always manufactured under GMP guidelines. As I said, GCP guidelines for clinical trials, all the IPs which we are undertaking a clinical trial has to have a GMP guideline certificate as well. We can only do a preclinical study with the R&D batch data. But as soon as we move to a phase 1 clinical trial, we have to ensure that the product or IP has to have a GMP certification. Only preclinical can be done by R&D batches. We have to have systems that produce quality of every aspect of the clinical trial. So these are the 13 principles of GCP which we have to adhere to while doing clinical trial. Now there are a lot of chapters in clinical trial, from sponsors' responsibilities to investigator responsibilities to IP dispensing, making, producing, and also the records as well. I will just touch upon the investigator responsibilities. So the investigator should have a qualification, you should have an up-to-date CV, compliance with GCP, permission of monitoring by sponsors and regulatory authorities, maintenance of list of qualified personnel who are delegated for each responsibility. Suppose you have a CRC at a site, you have to have a delegation log which says that XYZ is a CRC, ABC is the pharmacist, OPQ is the nurse. So you should have a duty delegation log which says that each responsibility is being done by a specified person at a site. The investigator should have adequate resources, it should have enough patients to recruit, it should have enough time to conduct a study, they should have a qualified staff, they should have enough facilities, and also they should be trained to do a clinical trial. A qualified physician is responsible for the trial-related medical decisions, provide adequate medical care for AEs or other significant medical conditions, inform PCPs about participation in a trial, make a reasonable effort to ascertain why participants withdraw from the study. Suppose a patient withdraws from a study, an investigator should try and find out that why did he really withdraw from the study. Was it an adverse event? Was it that the visit schedule was very difficult for the patient to come? Was it that there was lack of efficacy? So we as investigators should find out why the patient dropped out from the study and also document that as well. Everything. So whenever a patient goes out of the study or removes his participation, we have to find out that why did he really get out from the study. Communication with IRB. This is very, very important. We always feel that the communication with IRB has to be done by the sponsors. No. All the communications with the IRB or the ethics committee has to be done by the investigator. What does the sponsor do is, a sponsor supports you in terms of getting the documents ready for the submission to the ethics committee. But all the documents, if you see, goes from your letterhead. Your letterhead goes asking him to do a clinical trial and approval. Your CV goes, your undertaking goes, your MRC detail goes. All these details from your side goes to a ethics committee for approval. Sponsors can only support you in terms of the ethics committee fees as well as the dossier submission to the ethics committee as per the requirement. So if you all know this, every ethics committee has a different set of requirements for their EC approval. Some say that they require 5 copies, some say that they require 10 copies, some say that they require the financial agreement as well, some say they don't require. So every SOP is EC is different and every SOP requirement is different for EC approval as well. So the communication between the IRB, the ethics committee, is to be done only by the sponsor. Suppose there is a AE or adverse event or adverse drug reaction at your site, you as an investigator will have to inform your ethics committee that such an adverse event has occurred at your site, nor the sponsors. It is very important that the investigator complies with the protocol. There could be protocol deviations or violations which is okay in doing a clinical study, but you have to see that you adhere to the protocol and its requirements. So a proper training of the protocol is very, very important when you undertake a clinical study. IP accountability, also important. I was doing a drug on oncology where the IP, each tablet cost about 20,000 rupees. You know, for a 6-week therapy and all, the entire therapy was for 36 lakh rupees. So IP accountability and storage is also a very, very important factor for an investigator when he undertakes a clinical trial at a site. How many medicines were being used, how many were destroyed, how many were unused, how many were returned back. So all these are also a responsibility of an investigator when he undertakes a clinical trial at his site. Again, as I mentioned, all the records have to be stored, all the source data has to be stored. You have to store your data for, especially your intervention study, for 15 years after the product is being launched in the market as well. So ensure that whenever you undertake a clinical trial, what happens to the, not only the CRF form, but also to your source data, your X-ray frames, your MRI records, your lab reports, all those have to be stored for a period of 15 years, especially for interventional trials. And hence, whenever you conduct a clinical trial, you have to first discuss with your sponsors that what happens to my source data, where does it get stored, do I store it, do you have a third party where it can be stored? So all this has to be discussed before a clinical trial is being undertaken. As I said, again, the progress report has to be informed to the ethics committee on a regular basis, maybe 6 months or yearly. It all depends upon the ECSOP. Suppose there is an adverse event happening at the site, the sponsor has to inform the ethics committee about the adverse event and what treatment was being done. And maybe if it is SAE or something, the EC may communicate what insurance has to be paid or what compensation has to be paid to the patient. Again, if you want to terminate the study at your site, as an investigator, you have the rights to stop the study at your site and premature terminate as well. Again, as final reporting, so whenever the study gets over, you as an investigator will have to inform the ethics committee that the trial has got over. So these are, in short, the responsibility of an investigator while doing a clinical trial. We have many other responsibilities of sponsors, etc., which we can discuss later as well. Thank you. Any questions?
Good afternoon, everyone. Any questions regarding the ICH-GCP talk that you just had? I know it's a difficult talk to have post-lunch, but then, yes, this is one part which is very, very important if you want to become an active clinical researcher. So, yes, we would like to thank him. Thank you, sir, once again for taking the trouble. And yes, it was an excellent talk. But yes, believe me, all of would have had multiple questions if we were getting a certificate out of this for the GCP. Okay, so thank. Yes, I think. I just drop my contact details to the office. You can contact anytime whenever you require. I'm sure we can talk to Dr. Manohar and see that people who want to get GCP certified can be done by a simple training session on ICH-GCP. Thank you so much, sir. Thank you so much. Moving on, thank you once again. We will be moving on to the next workshop. First of all, I would like to thank KRSSDI for giving us an opportunity to, first of all, come in a room which has been named after our guru, Dr. Narasimha Shetty, whom we lost recently. And second, the person who is responsible for giving a push to many of these institutions, organizations, Dr. C.D. Srinivasmurthy. So I'm sure Dr. C.D. Srinivasmurthy, is he here? Okay, I think he's downstairs. But yes, a few lines of for Dr. C.D. Srinivasmurthy. He is one of the most loved teachers who was associated with DNB teaching programs across Karnataka. He has published several hallmark papers, both in national and international journals. He was associated with IMA, API, Holdsworth Memorial Mission Hospital, and was instrumental in building the MRCI hospital. So a big round of applause for Dr. C.D. Srinivasmurthy for his kind-heartedness. Starting with the diabetic foot workshop, yes, I know many of you have come here for the practical tips from the boss who is standing there, Dr. Sunil Khari. Many of you will be knowing him. My name is Dr. Pavan. I'm also a diabetic foot surgeon. I'm basically an orthopedic surgeon. I take care of the Department of Podiatry at Karnataka Institute of Endocrinology and Research. So when we were initially thinking of doing a diabetic foot workshop, and I was talking to Dr. Sunil sir, the whole idea was we did not want to talk about dressings. We did not want to talk about WAC, because knowing that you are all physicians, I mean at least 90% of you are physicians here, we wanted to make sure that we went back to basics. So that was the whole idea which originated, and that's how we started thinking of, okay, let's try to get all the physicians back to basics to see how well can a physician identify a diabetic foot, identify a pre-ulcerative lesion, and what can he do best with the setting that he is in. So that was the whole idea of this diabetic foot workshop. Yes, I know Dr. Shekhar sir was supposed to be chairing this workshop. I think he's just finishing one of his talks in the hall below. So what we would do is, we'll be going ahead. I have already given you the introduction of Dr. Sunil. Dr. Sunil, if you can please come. We will be starting off by giving small talks, and then we have a patient with us who is come from very far away. A big round of applause for her, because getting doctors to such programs itself is difficult. You can imagine getting a patient here. So we would really like to thank you, ma'am, that you took time off and have agreed for us to demonstrate certain things on her. Thank you once again. Dr. Sunil, all yours.
Thank you, Pawan. So at the outset, let me thank the entire team of KRSSDI. They just gave me this opportunity to share my experience and the company, what I have built up, Mandakini Medi Inch Health Private Limited, where we have taken up this first initiation, wherein my concern will be how best, as a surgeon, I have seen in last 25 plus years, varieties of cases. Now the issue is, it's only going to be a high risk, and high risk. So it's becoming a puzzle to most of the physicians. So I thought that let us solve this puzzle. You all are quite capable of handling it. I don't want any surgeon to work on this area. Let us work on basics. We are not interested in dressing. We are interested in preventing first ulcer. All physicians are interested in preventing first ulcer. So I have created a kit with this background, where it will be given by my executives are there. Devraj and Dominic, please create a kit to everyone who is there in this hall. And the kit contains the most important 2023 calendar. The first January, February, March will boost you that I am going to do this. I have given you a forefoot ulcer heals in 100 days, a midfoot ulcer heals in 100 days, a hindfoot ulcer, which is very difficult to heal, heals in 100 days. After that, after April onwards till December 2023, I have given you, please go through what you should start working in your clinic tomorrow, that is on Monday morning onwards. All protocols, guidelines, info cards are there in that calendar, and I have made particularly that April onwards, you will not appreciate any dates or anything, but you will appreciate only what you should do with the foot. I have given some pamphlets which should be circulated to all your patients, what education they require, 30 mantras, how I can prevent foot problems. Then the most important thing I have given you is a nail filer. Please make it a point that every patient should have a nail filer. He should never cut his nail. The moment he takes a bath, it is wet, ask him to file the nail, and everything ends there. Never allow any patient to cut the nails. So these are basics what I thought that I should reach to every physician and convey this, and let me work. The goal what I started with, as Pawan told, this was my concept, that our goal is first to Karnataka, rural diabetic foot should be forward to prevent world's cheapest artificial limb, that is Jaipur limb. I don't want anybody to have a Jaipur limb. That is how one has to work on. Every physician can do this. This is our goal, and we can achieve this provided the major role is from the physicians. Number two, the theme is what best 360-degree care can be given to prevent the first ulcer. And this can be achieved by preventing, by assessing every aspect of the foot, and how one has to deal with it, I will be dealing with in a short while. Everyone is looking at amputation. Everyone looks at the data. By the time I finish this 1 hour lecture, I think it is 60 minutes. And as you know, every 20 seconds, one limb is amputated across globe. So around 180 limbs will be amputated, or 180 amputations will be performed by the time we finish this workshop. So what is required? We need to address whether I'm going to cure this condition, whether diabetic foot is curable, number one. Number two, what is the number? Why you are so worried about it? Is it a big number, what we are seeing it? What is functional limb and what is non-functional limb? What is footwear science? Why we have not been taught in our curriculum in medical college that footwear science is very important now. Number four, what exercise advice I am supposed to give as a physician to the diabetic foot person? All of you are so much curious. I think morning they had a discussion on yoga and exercise. So what exactly we are supposed to advise to the patient who has a foot issues? Now we need to make connections to sort out the puzzle. What is that connection? We are unable to pick up at risk foot, we are unable to detect high risk foot, and we are unable to detect very high risk foot. So we need to have understanding regarding foot behaviors in diabetes, number one. And number two, plan to salvage. How you can salvage it? If you understand the behavior of a foot for a trivial injury, yes, you can salvage that limb. The next point was, we need to visualize the differences when I should refer it early to the next level of a higher center. As you know, metabolic, you are the leaders. Vascular, there is a person who deals with it. Microbiology, very important. Wound care is very important. Education to the patient is important. But the most important is offloading, offloading, and offloading. If you don't understand this new science of offloading, how you should offload the body weight when patient starts walking, I don't think any wound will heal. In spite you have an excellent coverage of all these. So we need to fix all these in the right way, then only the puzzle can be solved. What is our plan? Today's plan is, we have our strategy, and definitely we want to know what are the new challenges diabetic foot is going to come up with, what will be the new clinical presentation in next coming years, and what is going to be your new prescription, what you are going to give to the patient in coming years. It's very important now. And this is what all of us are trying to do to keep the foot in remission. Stay connected. How can doctors and patients reach us? It's an emergency. It isn't a foot attack. All of you are aware of heart attack, but nobody is aware of foot attack. Let it be acute, subacute, let it be chronic, let it be active or inactive. You need to get connected. And that is how we have created a DF helpline. Just connected on WhatsApp. Keep this number always with you. You have any issue anytime, just share the details to me. We are there to help you out. And this is how we are planning that we best reach across rural Karnataka to prevent amputations and to prevent Jaipur limb. Thank you so much. I think I am supposed to request Professor M.A. Shekhar sir to talk on a very, very important topic, how a glycemic control can prevent an amputation. I think sir will join us later. Meanwhile, I'll ask Pawan to deal on nail care and skin care for just 5 to 6 minutes. Thank you.
Thank you, sir. As usual, a small talk, but he conveyed a lot in that. So before we begin, I think Dr. Shetty Srinivasmurthy sir is here. Thank you once again, sir, for your big-heartedness. A big round of applause for having supported KRSSDI through difficult times. And yes, we will be trying to make sure that your name lives on in the form of all these programs that KRSSDI does regularly to make sure that our staff, our medical staff, paramedical staff are updated on the knowledge not only regarding diabetes but also the complications of diabetes. Thank you once again. So yes, coming to my presentation, my work is very, very difficult now because Sunil sir has given me only 7 minutes to talk, and he has given me the topic of skin and nail. I mean, we all know skin is the biggest organ, right? Largest organ. But then what I will be trying to do, yeah, thank you. So what I will be trying to do is, we will have an agenda just like Sunil sir had. We will try to see what are the different reasons for increased incidence in diabetic patients, what are the types and the treatment in a very, very small way, and how can we prevent. The cool mantra today is prevention. We are not looking at treatment, we are trying to prevent them, okay? So coming to introduction, very basic, skin disorders, almost in 70 to 80% of your patients, you will see skin disorders, especially in the lower limbs, and nail disorders almost in 20 to 30%, and in fact more if the patients are coming from a poor socioeconomic status. So the most common changes, what are the reasons? I will not go into the details of these things because you guys know this much more better than us. What are the reasons for the skin changes? What are the reasons for nail changes? Poor hygiene, poor socioeconomic status now cannot be a reason for the same. Please take it that I have seen people coming from slums who have maintained their feet in a better condition because they were ready to change their attitudes. So attitudinal change is the one that we have to start targeting. So again, reasons for nail changes, periangular erythema, capillary alterations and glycation end products which get collected in skin, the superficial areas of skin as well as the nails which cause all these. Now there are different types of problems, basically divided into 3 types, associated with but not specific for diabetes, due to diabetic complications, and associated with neurovascular complications. So coming to the most common ones that you will be seeing in the lower limbs, the most common that your diabetic patients will show is furunculosis. We all know this. Hair root infection, which can happen very, very common in wet conditions. So most common is the staph aureus, starts as a tender area growing to 2 to 3 centimeters, and then central separation. Simple antibiotics and wound care will take care. Carbuncle is nothing but a collection of all these furuncles. But this, many of them think that it's just an incision and drainage. No, you have to explore this cavity to make sure that all the septations have been removed. Intertrigo, again, almost 80% of your patients will have intertrigo if you are able to look at their feet. So this is one of the most commonest things that I tell my physician friends, that the easiest way to not miss a diabetic foot, ask the patient to leave the footwear outside your consultation room, tell him to come in, see his feet when he's walking in, and by the time you have finished everything, put your hands on the dorsalis pedis and feel for the pulsations. Dermatophytosis, again, can affect web spaces, but yes, nails, when it affects the nails, is called onychomycosis. The problem with onychomycosis is the treatment takes a long, long time, takes almost 3 to 4 months. Dermal and epidermal manifestations, again, believe me, when I see, I am at a tertiary setup where many of these are referred patients, and almost 90% of my patients already have dermal and epidermal manifestations. So diabetic dermopathy, this is one of the most commonest ones again, but most very, very benign because these are atrophic hyperpigmented macules on the skin due to minimal trauma, mild pyoderma and or folliculitis. They are irregular, rounded, oval, and they do not cause any problems. There is a high correlation with retinovascular disease. So that is one point that the physicians can pick up when you see a patient with diabetic dermopathy. These will not be itchy. They will just be there. They will not be itchy. They will not cause any problems to the patient. Diabetic bullae. How many of you have seen diabetic bullae? Yes. So this is how common diabetic bullae are. Please do not put it on the patient that you have walked barefoot, you have put pressure on it, and the patient will vehemently tell you that, no sir, I have not done any of them, because diabetic bullae do not happen because the patient has walked barefoot. They are common in lower limbs. They can be spontaneous. And there are reasons. There are people have looked at by taking a biopsy. They have found out there's some problems in stratum corneum causing it to become a bullae. But basically goes to say that these bullae are usually not pus-filled. They will have sterile fluid. The only thing that you have to do is puncture them with a sterile syringe, leave the skin there, do not try to deroof it, but in case when you are puncturing it, if you find out there is purulent discharge, please deroof it properly. That's not a diabetic bullae. So again, there are 3 types: spontaneous and non-scarring, hemorrhagic and scarring, and multiple tender non-scarring. How many of you have seen ingrown toenails in your patients? Or many of the patients? Yes. This is how common it is again. The predisposing factors are poorly fitting shoes. This is the most common cause for an ingrown toenail. Improper nail trimming, which we will be trying to demonstrate on Sujata today. Excessive sweating, that can be a cause. Nail infections and nail apparatus abnormalities. And how can we avoid? Proper footwear, wide toe boxes or open toes with proper cushioning or proper protection. Proper trimming of nails straight across. What do you mean by that? I will talk to you in the next slides. Addressing the underlying pathogenic factors like treating hyperhidrosis or onychomycosis. Cleaning with hydrogen peroxide or iodine. This is doubtful, but yes, we will show you. Silver nitrate application for the granulation tissue. Why am I talking about granulation tissue? Because these are the 5 different stages that your patient will come to you with. The first stage where it looks almost like a normal nail, but the patient will tell you that he is having pain because of local irritation. Stage 2 where you will see a proper paronychia, there will be an abscess. Stage 3 where there will be a nail lysis at that area, you will see some unhealthy granulation. Stage 4 where there is the nail would have come up, the nail lysis would have started, and stage 5 when it is bilateral. So we again have a different, what our idea is to understand that if you see this, you have to tackle this patient, because otherwise this itself can give rise to a great toe amputation, a hallux amputation. So don't try to neglect it by just giving antibiotics or by just cleaning it. If it is not settling down, it is time that this patient goes on and meets one of the specialists. So what is proper nail trimming? Now you see 4 ways in which this nail can be trimmed. How many of you think that A is the proper nail trimming? None. So how many of you think that B is proper nail trimming? How many of you feel that C is proper nail trimming? Still none? So none of these are proper nail trimming? Yes, D. So everywhere we keep hearing straight across, straight across. What do you mean by straight across? I don't know whether you people have done it straight across. You try to do it, it's difficult, because every time we try to go inside and try to remove that extra part of the nail which you are trying to see, which is visible here. We think that this is the extra nail and we try to remove it. In diabetics, don't do it, because whenever you are trying to do that, the chances that you are injuring the underlying substance there is very, very high. So that is the importance of straight across trimming of the nails. Importance of a footwear assessment. How many of the physicians here assess the footwear of the patient? Very less. So this is one case which is going to demonstrate how important it is to look at the footwear. Now this lady, elderly lady, around 55, 58, she came to me, very good control of sugars, and was sent to me by one of my colleagues at my institute saying that the lady is following all the things that you have told her. She's wearing footwear, MCR footwear, which she thought was proper footwear, MCR Hawaii chappals, and she says that this wound has started since 1 month and it's not improving. So when we took her up, this was what it was. This was a callosity. Any callosity in a diabetic foot patient, consider it as a pre-ulcerative lesion. So that was the time when we decided that let's have a look at the footwear. And this was the footwear that she was wearing. Okay? This is not the proper footwear, but okay. Let's go to the next slide and see what is happening to this. You see that? Every time a neuropathic patient is walking on this footwear, which has an embedded stone in that, patient will not feel a thing. And that's the reason why the patient is coming back to you, thankfully with only a callosity. Can you imagine this patient wearing this and walking more than 100 steps a day for the next 1 month? This patient is going to definitely come back to you with a wound, with an ulcer. So one of the first steps of preventing the first ulcer, please look at what the patient is wearing. So we just removed it, told the patient to come back to us, everything healed. The importance of avoiding extremes of temperatures. I know many of you would have, I know there will be the mantras in the things that we have given you, Dr. Sunil has given you. Avoid extremes of temperatures. Forget extremes of cold. We rarely get extremes of cold in our place. Extreme heat, that is one thing. Many of your diabetic patients will have pain-filled peripheral neuropathy. And for that, they will be using electric blankets, hot packs, dipping their feet in hot water with salt. So many of you, if you are telling them, please stop telling them that. Do not tell the patient, a diabetic patient, to dip their feet in hot water. Does not help in any way. It only causes further problems. Now this patient is again an elderly man who came to me. I'm so sorry, I don't have the exact preoperative images wherein there were bulle and all. So the patient was going to a bath, and without checking the temperature, he just took 1 mug of hot water and poured it on his feet. And he came back with this. So it is important that you educate your patients how to prevent these. These are very easily preventable. They can check the temperature, not with their hand, because their hand would also be involved. Tell them to use their elbows. It's very easy. It hardly takes 1 minute, but that is going to prevent these wounds for you. Okay? And sadly, the right foot, this was the regular follow-up, and we could not save the left, second and the third toes, but thankfully the right became absolutely normal. It just goes to show how simple things, how simple education will improve and prevent the first ulcer that the patient will develop. So with this, I will finish my talk. Yes, there are so many pamphlets which we have been making. We have been trying to do so many programs, educative programs. We recently did something called as a Safe Feet, Safe Ride, wherein we went to the PHCs trying to raise awareness about diabetic foot. Every one of you can do it. By the time the patient leaves your consultation room, you could have told them at least these 9 to 10 points. Give them a flyer, give them a pamphlet, at least let them know what they can do to prevent that first ulcer from happening. Thank you. Yes, we have Shekhar sir with us. So, no problem, sir. So we would like to hear from you.
Right. I'm sorry I was late. I don't know how it happened. 2:40 I was supposed to speak, and 3 o'clock I was supposed to be here. It got, all these things happened. Now, Dr. Sunil Curry, who is a very good friend of mine, his name is Curry, but I am skin-wise, whatever you want to call me as, okay? He called me and said, Dr. Shekhar, you should be, actually he said, sir, I'm sorry, he's as good as me and I am as good as him. You must be a part of the diabetic foot workshop. And I said, what do you think I will bring to the table to the august body of what I should speak? He said, talk about prevention. I readily agreed, it's very easy to prevent, etc. like that. When I thought about it, I came to know it was a Pandora's box. You know the story of Pandora, the Greek semi-goddess? The king of gods gives her a box and says, don't ever open this. And inquisitiveness takes better of her, and she opens up all problems of the world, and she is unable to put it back. That's why we are suffering, it seems, Pandora's box. And abdomen, you all know, they say in medical this thing. And now, put the brakes on diabetic foot complications. He has given the topic. This is my disclosure. Fact, everything we hear is an opinion. It's just an opinion. What I'm going to speak is an opinion. Pawan, Sunil, it's an opinion, but it's an educated opinion. But it's not a fact because it keeps changing. And truth, everything we see is a perspective and not the truth. Keep this in mind. Fact and truth is the thing, disclosure that you should have. I don't know, I went to thank you very much for your patience. Deliberately I put it because it's such a patient hearing when a stupid man like me speaks to you about diabetic foot, which is not my field at all. Anyway, I remember in my early days of practice, when I was young, physically, not mature enough mentally, many of our patients, we used to tell them, "Ang madbadi, don't do this, don't do that, it will happen," and all that. How to prevent? An elderly person said, the biggest way to prevent is not to be born in this world. If you are not born, you can prevent a damn things. But we have already taken birth. How to prevent? The next biggest step is to prevent diabetes. Impossible. So, we are now in the problem of how to prevent diabetic foot problems, diabetic kidney problems, diabetic that problem, this problem, because we have diabetics in front of us. A very, very practical, pragmatic question that we have to answer. This one, I hope, AHA 2010, American Heart Association, came up with this. Simple 7 concepts. Reduce blood glucose. I should have written glucose, it's written sugar. Sorry for that. Control cholesterol, manage blood pressure, stop smoking, get active, lose weight, eat better. 7 concepts. Are you doing it? Say yes. If you are not, stand up on the bench. This, they called it as life's simple 7. Simple preventive measure to prevent your diabetic or yourselves from having a heart attack, having a kidney failure, having diabetic foot, blah, 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 is life's simple 7. Can we do this? If you can do it in your clinical practice for your patients, you can prevent it. I can stop here, but I cannot. I'll go for a few more slides. Barriers. Unfortunately, these two are the barriers. The community and the system we live in is the biggest barrier. Whatever you tell them, you have told them, lost in translation, as Pawan said, please wear footwear. But we don't tell them what footwear, how to wear them, when to wear them. This is what he was talking about. That is the system barrier. Community barrier, no, we can't wear chappals into kitchen, we can't wear chappals into that room, this room. All those population admixture, etc. And personal barriers. The biggest is poverty. How do you define poverty? Empty pockets. No, it is poverty in their intelligence also. Please keep in mind the biggest barrier from the patient side and also probably from the doctor side is paucity or poverty of their thinking process. These two barriers, if we can overcome, we can prevent it in a big way. Factoids, we see so many Google scholars, Google University trying to give certification to all this. This is another major thing. Factoids are semi-truths, not full truths. These 3 barriers, if we can overcome, it's not very easy, it's pretty difficult. Now, why do you think diabetic foot is very important? You don't have a heart, can you live? No. If you don't have one kidney, probably. You don't have brain, definitely. But if you don't have a heart, you cannot live. You don't have a foot, you can probably live. You don't have feet, it's very difficult. But from a diabetologist perspective, from a diabetic foot surgeon's perspective, diabetic foot is very, very important. Why? For heart it is macrovascular disease, for nerves it is microvascular disease, for retina it is micro, but diabetic foot is both macro and microvascular. So if you can prevent a diabetic foot, take it from me, you can prevent cardiovascular, retinopathy, neuropathy, and to a certain extent nephropathy. Can you understand that? That is the importance. And thanks to pioneers, that young Turk there and this old Turk here, thanks to them, they have brought to the forefront the importance of diabetic foot. Take care of diabetic foot. Prevent them from primary prevention or whatever it is. Take it from you. You can prevent a stroke, you can prevent a heart attacks, you can prevent heart failures and probably kidney failure. Right? You all agree? How do you say yes in India? You say yes in India like this. How do you say no in India? So we have to change our mindset. Like diabetic foot, you have to change mindset. Say yes like this, say no like this. Similarly, take care of diabetic foot, you will take care of micro and macrovascular complications. Prevent diabetes. I think these are all theory. Okay? You can all read about this. But what's important is, you have taken the step in preventing all these things. How do you prioritize? What is the priorities in prevention and management? I will not touch. I leave it to these two people to do it. What is the first priority to prevent diabetic foot you take in a diabetic? Anybody? Regular inspection of the feet. Glycemic control. Fantastic. I think you are doing a great job with your diabetic patients. How many patients? Wow! Give her a hand of appreciation now. The first step in a diabetic you take is blood glucose control. Inspect the foot, it's all right. Right? First step you have to take is glycemic control. How do you do glycemic control? It's beyond what we talk about it, lifestyle modification, education, euglycemia, etc. like that. But the next step is blood pressure and dyslipidemia. What is dyslipidemia? For a diabetologist, it is LDL below 25, 75, 100. For a diabetologist, LDL below 100, HDL above 45, 40. For men, 50 for women. I don't know why women should have more. The rest it's fine, okay? But for a cardiologist, crazy people, less than 50, LDL. HDL, more the merrier, more than 60, it's impossible. But proper blood pressure control from 160, 90 to 150, 80, is it good? Absolutely. 5 millimeters systolic can bring down 2 to 3 times the macrovascular disease. Simple. One step at a time. So we will not look at the guideline cut-offs. We look at bringing it down in a proper manner. And others, what are the others? Deliberately I put it, you can fill the blank any which way you need. Anything else. But the first step, as our great dietitian said, is blood glucose control. Whoever it is, young, old, middle-aged, whoever type 2 diabetic or whatever, blood glucose control is paramount. Then blood pressure, lipids and lifestyle, etc. like that. I hope I made myself clear. And lastly, look at this. The conundrum. Non-veg fit and active. All those puma, leopards, tiger, they are fit and active. They eat non-vegetarian. How many of you eat non-vegetarian? Lot of people. So you are fit and active. Except this guy who is a... just joking. I know him very well. Okay? Look at the other side, the vegetarians. Elephant. They were saying, a doctor was saying, no animal is obese. I think he has not seen elephants. I think he has not seen a Gendamruga, rhinoceros. Okay? They are obese. Probably not. You have seen a Gendamruga, rhinoceros. The fastest speed it can reach is about 30 kilometers per hour. Beyond that, it can't run. It can run only for 50 meters and then it stops. Sorry, I can't run, Shekhar. I'll stop. Look at this. What is the thing that you should get? Because let's not confuse our diabetics about unnecessary diets. Non-veg versus veg, etc. The reason is, it's impossible to reduce weight by eating green grass and salads and walking. Both non-vegetarians who are very active, vegetarians who eat salads, it's impossible to lose weight. You have to shift your gear for weight loss, because I was told by Sunil, who is more of a physicist, he's a physics expert more than a surgeon. Stupid fellow. He doesn't look at diabetic foot just for the sake of vascular, and he also looks at the kinetics of the bones and muscles. Great activity. This is it. Okay? It's not just walking, it's not just eating, it is something else beyond that, which we have to decide. I'm sure Sunil will give you a very good idea. At the end of the day, patient-centered approach. I have deliberately put only these 5 or 6. Impact on weight. Whatever you do, blood glucose control, blood pressure control, dyslipidemia, you must look at the impact on weight, because 1 kg weight gain, 12% incidence of heart failures, 6% incidence of heart attack, all those funny things we have read about. But when Sunil told me quite some time ago, many years ago, 1 kg weight gain puts 1.5 kg on each lower limb. So 1 kg weight gain puts 3 kgs on the feet. So if you think blood glucose is fantastic, blood pressure is very good, dyslipidemia is normal lipidemia, but he has gained 5 kgs. Take it from me, they will come with calluses. That's what he was telling. I fully agree with you. Patient preferences. Chapli akadala sir. What does Pawan do? Hey, voya tala tinbada. So you have to have an answer for that. Any answers you have? Anybody? ಈಗೆಲ್ಲ ಗೂಗಲ್ನಲ್ಲಿ ಸಿಗುತ್ತಲ್ಲಪ್ಪ, thick socks. They prefer to wear socks. A little bit thick sole, socks they can wear it inside. Outside, whatever, simple answers I'm giving you. I'm not an expert. Cost, efficacy, etc., etc. You should think of. Okay? What happened? I don't know. Something wrong. Okay. Do you understand now? Deliberately I have put all this. Otherwise, patient-centric approach is patient in front, all doctors and nurses and all those people in and around. No. When it comes to diabetic foot care, it is the patient preferences first and then doctor's expert opinion. So this is my last slide, thankfully. What happened? All of us have gone through high school, almost done. We went on for the first 10 years of our existence. Then what happened? We went into PUC. Then we went into medical college and we went to the top. Is that the end? It's only a small peak that you have gone through. So OSHIT is the first thing that should come to you when you see a diabetic foot. Thank you.
Thank you, sir. Thank you so much. Thank you so much for putting the radar back on biomechanics of a diabetic foot. I'm sure Dr. Sunil will be touching on that. He was our director some years back, and we used to just love presenting to him so that we get the questions. And these questions usually made us think. And just like what you people are thinking now after going through all the slides that he has just shown you, believe me, this workshop, I hope, will make you think. Over to you, Dr. Sunil.
Well, the next presentation, what I'm going to show you is what is a 360-degree care, what we need to render through physicians to prevent the first ulcer. (360-degree care). Yes, yes, correct. See, I gone to the a 360-degree care to prevent the first ulcer. Very, very interesting. I could get this beautiful template where, as you all know, that I have been working in this area since quite a long time. And I love regarding foot and ankle biomechanics, which is very, very interesting, very challenging. And I love to design footwear for all high-risk feet. High-risk feet is the one where it's a greater challenge to everyone across globe. And I've been with so many societies. Especially I love to be with International Foot and Ankle Biomechanics community, where it has taught us so many new things. Every patient of diabetic is something unique. From roots to results. So let us go to the basics. In Bangalore, in Karnataka, whether diabetic foot is happy, is it cared, is it safe, is it functional? Or is it lying in one of the corporate hospital months together? Or is it lying in the house months together? Basically, lower limbs are organs. They are organs of ambulation. If they are not functioning, and you say that I have healed a wound, I don't think a podiatrist surgeon or any surgeon has done a great job on that. That limb has to walk. It has to ambulate. Then only your job is complete. Now, what was there in last 35 years? The first conference on diabetic foot was done in 1986 in Melbourne, where in UK they could see the longevity of life of 85, 95 and 100 plus because of use of insulin, and they noticed foot problems in their patient in 1986. First conference on diabetic foot was conducted in UK, Melbourne. Since then, in last 35 years, what we have learnt? You just go retrospectively. What we did well in last 35 years? We came to know it is a common, complex and costly affair. Yes. Highest recurrences, highest relapses, highest failures. The best of the best centers in the world. What are the parameters to say that I have done a good job? Evidences. So we have got two evidences in diabetic foot. One is a clinical healing of an evidence, second is a radiological evidence, which is not there radiologically in any medical departments anywhere. It is only in diabetic foot. If you want to say that I have done a good job in a foot, then you have to have two evidences. One is a clinical wound has healed, and take an X-ray and prove that there is no bone involvement. Then only you are right. You can't say that only wound has healed, so you have done a good job. What didn't work in last 35 years? We utterly failed to prevent the first ulcer. Utterly failed. We are struggling to heal the ulcer. We are using number of methodologies, number of dressing materials, number of advanced wound care modalities. And we miserably fail in preventing the recurrence. More than 40% they recur within 1 year. So callus, pre-ulcer, ulcer, heel ulcer recurrence have been a challenges. So a short overview, I am going to talk on cure, myths and facts, where I had created one group, diabetic foot workshop group, where they kept on asking me, and a lot of suggestions came. What is functional foot? What is non-functional foot? What is footwear science? What is exercise advice a physician should give to a diabetic patient? What are the new challenges? What is the take-home message? I cure diabetic foot. Wrong. You can't cure a diabetic foot. So what it is? There is only arrest. You can arrest a disease process by doing or advising the things. You can't cure the disease. Make it very clear. It is an ongoing process, and as long as he walks, he is going to break. What is success in diabetic foot? Success in diabetic foot is, sir, I have kept my patient ulcer free for 5 years. So as good as cancer. That is your success. Second, surgeon says, I have maintained a plantigrade foot, sir. Success. Shoeable, braceable foot I have given back. Excellent. I have kept foot in remission like cancer. I have not seen that after he went through ulceration, again I have kept the foot remission for 5 years. Excellent. That is your success. But don't expect that I am going to cure diabetic foot. It will never happen. And somebody says, even I have changed my consent forms to all my patients when I do surgery. There is no cure. I am not curing anything here. I am arresting a disease process for time being. This is a module, what we work on. This is a stairway of amputation by Rogers and et al., where it's very clear that your role is very important in primary care and secondary care. Tertiary care is altogether different. So we have different segments: normal, DF care, PNP, PVD care, DFU care, DFI care. Most important, rehab. I get many patients, sir, nan kaalvan tagdari, innond kaal sarl police kodavak. Look at the approach what patient come to me. I have lost one limb, sir, but I don't want to lose other limb. So rehab is so important. You have to work on that segment. Either you are working in primary care or you are working in tertiary care. Footwear science is very, very important. To prevent a first ulcer, to heal an ulcer, and to keep the ulcer in remission. It is a footwear and footwear science. One has to understand in a right way. So every patient who enters into your clinic is always with a question, look at my feet and tell the prediction. You just predict, sir, whether I will go through any amputation or something like that. So we have a stratification 0, 1, 2, 3. And all of you know, in 2019, we had a latest inclusion criteria of PAD. That means we missed. In so many years, we missed. And what is stage 3? If somebody is CKD, dialysis, you need not have to assess anything. He is already in stage 3, grade 3. So we missed all these people in last 30 years. The first guideline was in 1996, 2015, 2019, 2023. We are getting one more guideline now. 160 countries meet and they draft the guideline. So this is something new turn we had in 2019. PAD, peripheral arterial disease, has to be assessed. Then only you can say that he is 0. There has to be no LOPS, loss of protective sensation, and no PAD. Then only it is stage 0. Otherwise, no. Earlier we never used to check. It was all together. So now the things are totally different. Now what I want resource poor clinics, rural practitioners to know is, very high risk, stage 4 is barefoot walk. Overload footwear. Sir, you ಬೈತಿದ್ರಿ ಅದಕ್ಕೆ ಹಾಕೊಂಡು ಅಡ್ಡಾಡ್ಲಿಕ್ಕೆ ಚಪ್ಪಲಿದನ್ನು. Overload footwear, very, very wrong. Acute Charcot and chronic Charcot you are missing in your clinic. Very, very bad. It's a very high risk field. This is one classification. People are asking me that, sir, tell me a new classification where I can pick up the cases. YFE classification. Wound, ischemia, foot infection. These 3 criteria together, you can come out and predict, as you have a TNM classification in cancer, that whether this foot I can save or he is going for amputation. So these are very, very important things. You can go refer, you can go through it. The most important thing is, should we preserve this limb? One has to assess this with your surgeon. It's a multidisciplinary team, but in India, I wish a good physician and a good surgeon, a 2-member team can salvage maximum limbs. Can the patient walk in the community? Can he walk at home? Can he transfer his weight from one room to another? No, he is going to be bedridden. So is it worth saving his limb? So criteria to salvage are also very, very important. Foot screening, basic screening is PNP assessment, PVD assessment. Footwear assessment, you need not have any dynamic pressure mapping. You can just go through the footwear. Where 7-8 years back when I was invited there in Mysore, I think last session, all the patients were seen only when I saw, after sir's permission, I used to see first footwear and I used to tell this patient is having lesion here. Am I right, sir? So it is a footwear which will give you 80% of the clinical acumen. If you know footwear, how to examine, what to see, everything is written there in the footwear. Number 1, number 2, number 3, don't miss footwear examination. And if you are interested in diabetic foot care, then keep your CPR score very high. Because if you are caring your feet, diabetic feet, I just give 1 mark. I am protecting the feet, I give 3 marks. But if you don't know what to do next, you should know how to refer early, because that carries 6 marks. If you are not doing that right reference early, look at the timely references, everything, cost, morbidity, mortality is low, but a delay leads to amputation and death. At just 2 weeks delay, we have lost the patients. So everyone has to have a CPR score which should be above 6. These are the foot types, what you will be seeing in your day-to-day practice. But the commonest what you are going to see in next 10 years is this. Either somebody has got his grade 2 amputated, second 2 amputated, because everyone is going through high risk now. So to prevent this, we thought, why not get our colleagues, physicians into action now? They have to prevent the first ulcer. Then only we can prevent this kind of feats, which are going to be in big number in coming 10 years. What are the myths? Dressings heal the wound. Nee dressing madko dogpa, ad gayam aiti. No, it never happens. If that would have been the case, why so many people are wandering from place to place? So this is a myth. What is a fact? Dressings don't heal the wound. A properly selected dressing enhances your body's ability to heal a wound. A dressing never heals a wound. Number 2, diabetic foot ulcer is a medical problem. It's a myth. It's not a medical problem. It's a mechanical problem. Your body weight hits the ground, breaks the feet. What is a myth? Healing a wound, keeping wound healed throughout is the same science. If that would have been the case, why there is a recurrence in such a number? So healing a wound and keeping it healed throughout are two different sciences. You have to understand it well. The moment you look at an ulcer, you always say, Mulu chuchitta, Mudi chuchitta, Chapletto, Yen Badskondi. Wrong. This is not outside in injury. Nothing has come from outside. It is inside out injury. The metatarsal head, the joints, IP joints, they lose the cushions which are there in the diabetic normal people. They are lost, pushed forward or thinned out. And whenever he walks, the repeated moderate forces hit the ground, break the skin. That is how they come to you. But you keep on asking them, Mali chuchittu, Muli chuchittu, Kal chuchittu. No, it never happens. It's 80% of the ulcers are because of repeated moderate forces. What are the clinical types? You know, pre-ulcerative, ulcerative, non-ulcerative, which is very important. I want physicians to pick up these non-ulcerative feet. Active disease, chronic disease, neuropathic, which was earlier thought 70%. Ischemic was just 30%. Now it is neuroischemic, which has taken 45%. Critical ischemic, acute ischemic, which you have seen in COVID. I have brought this new one for all of you. Renal foot, cardiac foot, HIV foot. HIV is no more an acute disease, it's a chronic. Necrotizing fasciitis foot. Psychiatry patient with diabetes and diabetic foot. Critical limb threatening ischemic. Such, I had all these varieties. The most difficult is renal foot. If anybody is on CKD, on dialysis, and you are handling it with a small ulcer there on the heel or somewhere, some part of the foot, it is not going to heal. It is the worst foot. What goes wrong? What is functional foot? What is non-functional foot? It is altered biomechanics in diabetes, which leads to sensory, motor, and autonomic neuropathy. The non-enzymatic glycation end products get deposited in all the capsules, tendons, and fascias. So what happens? You have a sensory insensate feet, you have a rigid feet. I don't know how many of you have seen rigid feet. It's a log. I see in my practice, there is no mobility of the great toe, there is no mobility of the ankle joint at all. Everything is fixed. He walks like this. And this is the worst stage. And I always tell all the young surgeons, never touch rigid feet. You can't change that rigid foot. You have to just get that rigid foot in a footwear. You design it in such a way that you heal the ulcer or prevent ulcer. You can't do any surgery. You do the surgery, it is again healing by fibrosis and becoming more and more rigid. So the rigidity, if it is there, try to work on a different line. So any rigid insensate feet, the moment it breaks, we call it as a non-functional. Functional foot never breaks. So what is basic foot concept biomechanics, as all of you know? Because of the insensate, the body weight gets fixed at a local area and maximum pressure is there. There is a callosity, and callosity is inversely proportional to the area and directly to the body weight. And that's how you get the break and ends up with the osteomyelitis. You have to look for the basic mobility of the great toe. If it is not, there is no dorsiflexion, that foot is going to break. If there is no dorsiflexion of the ankle, that foot is going to break. It doesn't require any gadgets. You don't require any n number of gadgets to deal with this, to check. Just take the great toe, try to mobilize it. Foot, try to do this. If it is rigid, that foot is going to break. So this is what we want you to people to assess in day-to-day clinical practice. What diabetologist can do? Callus, trim it. You have prevented ulcer there. If you are not trimming it, you are all afraid to hold the blade. Okay, fine. Today I am going to tell you how you can do this with a nail cutter, nail trimmer. I have seen, we did it with the USV DEF program, where we gave hands-on training, but 80% of the physicians were not willing to go ahead with the trimming of that callus. So then I thought that we should give them something new idea by which they can reach there. Preventing first ulcer in a unilateral limb edema. It's an active Charcot. What we did, we achieved inactive. We could bring it back to normal. How we did this? What are the steps? Weight bearing X-ray number 1, we could pick up fractures. Number 2, TCC, total rest, 1 month, 2 months, 3 months till the temperature comes back to normal of the contralateral limb. Then put the feet into the crow. This is a crow shoe. Fine, we could achieve this. What is happening today in day-to-day practice? The moment you see a unilateral limb edema, tablet Ditor, Lasix, 45 minutes walk every day. Review after 2 weeks. This is what the prescription I see with most of the diabetologists. So what is the end result of this? This is the end result. There is a midfoot ulcer. And you blame diabetes for that. Diabetes is not a culprit here. You should have given 100% rest, TCC, and put the feet back into the crow shoe. Then only the foot will not have midfoot ulcer. But if you write for unilateral limb edema, Ditor and Lasix, that is not a management. What is new thing? What is the new focus in 2019 has come for offloading? Whatever we were practicing to heal a wound. So that was a short-term offloading. Now the new concept is long-term offloading, till he dies. Lifelong offloading, once there is a break in the feet. So these are the new things, one has to go through it, and one has to do short-term for 100 days. I keep on telling 100 days are very important. If there is a break, it heals in 100 days. If it is not healing in 100 days, I am wrong somewhere. I have not done justice in management of the foot in a right way. Now, the next basic is behavior of the foot. Look at it, compromised neuropathy behavior. You see daily such kind of cracks, great toe at the heel level everywhere. When you trim it properly, there is a hidden ulcer there. This is a neuropathy behavior. But same patient comes back after 2 months, gangrene of the great toe. Then you blame diabetes for that. No, you have missed that ulcer there because you have not trimmed it. It is hidden there inside. This is a classical neuropathy behavior of the callus skin where you have got lot of hidden ulcers there. This is compromised vasculopathy behavior. Patient was told that there is an impending gangrene, please go ahead, meet vascular surgeon. She said no. There is microvasculopathy, nothing can be done, and we can't go, nothing can be done. My doctor has said, no, you can open up the vessels. That's the reason vascular department has grown such a level across India. And see, the end result is like this patient enters my chamber. They don't listen because they have been convinced by physicians that nothing much can be done. You can open up all the major vessels, macro vessels, where you establish the perfusion pressure, everything gets to normal. But this is not happening. So you need to understand there is only basement membrane thickening in the foot micro as such. If you open the macro, there is an increase in perfusion pressure and everything opens up. What is compromised limb and neuroischemic limb? This patient came to me with great toe ulcer. You were shocked how he could develop this. He said, I had been for cardiac checkup and the limb lead was kept here for 10 minutes. And simple pressure of that ECG has led to this ulcer. Look at the behaviors, how the foot is behaving. You can't blame diabetes for this. You need to understand the science, how it is going in a different way. A simple 10 minutes pressure on that area, he could develop an ulcer. And similarly, I have seen surgeons doing dressing on a sensory foot, tight, and that has led to ulcers on the dorsum of the feet. So you should never apply tight bandages on totally sensor feet. This is immunopathy behavior. Patient was admitted in a medical college months together, and he said it is totally now well demarcated. We will not do anything much. Patient came to me. This was extensive disease. We did above knee amputation and he died. This is classical immunopathy. A patient coming to you with 5-year, 10-year, 15-year, 20-year, 25-year-old history of diabetes, they have immunopathy. Anybody above 10, a massive immunopathy, their response is very poor for the healing purpose. This is footwear. Most important thing is to prevent ulcer, to heal and to prevent recurrence. Time to buy in the evening. Life of footwear, only 3 to 4 months. Diabetic footwear has a life of 3 to 4 months. Patients come and tell, sir, nimden factory tagadrenri, naak tingal navu tagobekandre. True, they have to change it. The most important part is the insole, which is 9 mm. If it reduces to 4.5, you have to change the footwear. If you are not doing it, he is going to come back to you with ulcer and he is going to have lot of problems. When to replace? Every 3 to 4 months he has to replace. What not to prescribe? How to prescribe in limited resource to a poor patient is very important. You can't say everyone that you should buy a footwear from such a company which is costing you 3,000, which is costing you 2,000 or 800 or patient can't buy, poor patients. So we need to find out some solution for that. What is it? What you look for in a footwear? Footwear screening is important. Look for the types of footwear, look for the wear, how he wears it. What is the insole quality? What is the outer sole quality? What is the size? What is the seam? Is it seamless or with seams? Adherence. Is he adherent to that? Is he using it regularly? Whether he has lost a great toe and is he using toe filler for that or no? All these are very important. Footwear science in diabetes. Foot doesn't know what to do in diabetes. Footwear has to do the job of foot. Foot doesn't know what to do in diabetes. Footwear has to do the job of foot. This is due to this kind of footwear. This is due to strap of the footwear. This is due to Jitendra style footwear where there is no enough toe box. How many of you seen in your clinic? 80% of the patient, I see them, they should come and sit in front of me and wear the footwear. They are using right into the left, left into the right. And that is the cause of the ulcer. 80% of the patient do this. We do it in childhood and we love that. And this is what happens when a patient comes to you, ask her to get the footwear inside, open up, see there is no insole at all at that site. And you want to give her all kinds of antibiotic. It's only 100 rupee good insole will heal that wound. I want you to know outer sole, there is a thorn, the most important size. I was shocked to see a number 9 on the left and 8 on the right side. I asked the patient, why you are with Adonki Bharat like this? He said, sir, ಇಲ್ಲಿ ಬಾವು ಐತ್ರಿ ಜಾರ್ತೈತ್ರಿ ಚಪ್ಪಲಿ ಅದಕ್ಕೆ ಇದನ್ನು ಸಣ್ಣ ತೊಗೊಂಡಿರಿ ಟೈಟ್ ಹಿಡಿತೈತ್ರಿ ಇಲ್ಲಿ ಸ್ವಲ್ಪ ದೊಡ್ಡದೈತ್ರಿ ಕಾಲ ಅದಕ್ಕೆ ಕರೆಕ್ಟಾಗಿ 9. See, they are so intelligent. They have started managing on their own because we are missing it. This is very, very interesting. So size, and this is what is very important. Customization of footwear is a new science, how we do in our center. See, he went through all this major surgery, he is with the footwear. So this is how we customize it. Not to have a recurrence, he is with this footwear since past 7 years. He doesn't have any recurrence. What not to prescribe? This patient came to me, I have an ulcer since past 3 years. I said, what you are wearing? This is what was given to him. There is a big hole. He said, who told you this? No, sir, hangada idre gaya maitad anta helyaru. No, this is not the practice. You should never do such kind of things. Most important thing is canvas shoes. Never look at those canvas shoes. Open up the insole, you see there is a very hard surface which is hitting on the plantar aspect. Check all these kind of soft top shoes which most of the patients are using. This is how I work in my center where 80% of my patients are poor. So I am a cobbler there. As a surgeon, I have to be cobbler and I want physicians to work in this line. When this patient came to me with this ulcer, and I just asked him to get the footwear, I could see rest all area was normal. It was only the first MTPG area which was thinned out to 3 mm. Then he said, sir, I can't afford anything. I have already spent enough. Then I said, okay, I just bought that insole which is costing 800 rupees. I just cut 1 by 1 inch. and placed it. See the healing. He paid me 10 rupees for that. It has done the job. This is how you need to reach to the poor. You have to reach to the poor. You can't say that he requires 800 rupees a new silicone insole. Why? There is no pressure anywhere else other than in that point. So why are you recommending that? He can't afford it. You can do this. It can definitely solve the issue. Increase the benefit, reduce the cost. That should be the motto of every physician and every doctor. Footwear to heal, as all of you know, forefoot, anterior wedge, midfoot rocker, and hindfoot posterior wedge. It heals well. And this is what exercise, which I have specially brought this slide, recommended exercise as per the guidelines of 2022. There are so many things written in RSSDI guidelines, but please define low risk, medium risk, high risk, and accordingly think what should be the number of steps per day. Foot, if it is operated, if he has an ulcer, please don't advise any exercise. Keep that infrared thermometer always in your clinic and check the temperature of the foot. If it is increased, tell him not to walk. Take rest. You can prevent the ulceration. All these foots, never ever recommend them any exercise. They require only static cycle or swimming if the wound heals. This is what guidelines has told you. I want you physicians to practice. An ulcer can heal with a proper plaster techniques, and I am going to demonstrate. I have not done any surgery. Everything has been healed, and I am going to demonstrate hands-on training. I am going to give you that you should start putting TCC. Need not be up to knee. You can have above ankle. Enough. I am showing you all those above ankle now. This is a non-plantar ulcer which has healed in 100 days, which was with PTV. Second, forefoot ulcer, we have done this in 100 days with simple above ankle TCC with a removable. We have made 2 casts, and then we have given him a footwear. He is absolutely fine. Midfoot ulcer, healed in 100 days with the same just above ankle TCC and removable it is. He can have a daily wash, bath, everything, and again put it back. Hindfoot ulcers, everything has healed in 100 days. Same TCC. This has to be practiced. There is no surgery done over here. There is just offloading done. And the most important thing, this has been recommended by Pawan and entire team, forefoot offloading and hindfoot offloading. When it should not be given to the patient, please tell anybody when it should not be given. Pawan, when it should not be given. Those patients who don't have proximal neuropathy, they have a proximal neuropathy, never prescribe them this. Distal neuropathy, prescribe them. If they are having proximal neuropathy, never ever prescribe. They have come back with fracture neck femur. All of them have come back with fracture. But the recommendations say you have to go for this. The recommendation have to be followed definitely for the distal neuropathy, not for the proximal neuropathy. Remember these things. These are the challenges where most of the patients. What is the new prescription you are going to write in next 10 years? Number of steps per day, not tablets or injections. You are going to recommend every patient number of steps per day because they are going to be highly sick. What antibiotic? Everyone is interested in antibiotic. If there is no clinical sign of infection around the ulcer, no antibiotic. No infection, no culture, no antibiotic. I have been seeing maximum MDR in last 10 years because you have been shooting antibiotics to every ulcer. Wrong. What antibiotic has to be given? You have to plan for acute. If it is neuropathic, always gram positive. If it is neuroischemic, always gram negative. Ischemic, always gram negative antibiotics. If it is a chronic, let it be anything, it is going to be gram negative always. What is the new challenge? You will be seeing both like. Already it has started in my clinic in last 10 years. I will show you that. This is going to be a greatest challenge. What offloading you are going to do? What you are going to plan with this patient? What quality life he is going to lead? This number is going to increase in next 10 years. Take home message, there is no cure. It's a temporary arrest. Ulcer-free days are your indicators of outcome. This is what is happening in my clinic. These two fellows are sitting with bilateral limb problems, and we are working on that. So we need a DF hotline. Please get connected. Please work on saving, especially rural foot. Very, very important. The way I have been seeing their sufferings, they have lost their entire family, all earnings, they have sold their fields, but outcome is they have lost both lower limbs. So we need to reach to the poor, and we should see that whatever the mission we have started now, I wanted it should start from Karnataka chapter, and I am looking forward. I think my good friend from Tamil Nadu is here, so I have requested him to pass on this message to the Central Governing Council that we should pass on our all care to the rural segment of India. Thank you. Thank you so much.
Thank you, sir. Absolutely brilliant. Yes, I do agree that we have to start educating patients. We have to at least tell them where to go for taking care of their feet. Yes, education is important, but accessibility is equally important. If you don't know, ask. If you don't know, if the patients don't know, tell them where they can go, what can be done. There are quite a few institutes now which are taking care of them, one of them being ours, Karnataka Institute of Endocrinology and Research. We have a very good department of podiatry. There are also private setups which are doing the needful, but as sir said, the importance of giving proper care at a subsidized cost to make sure that the patient is able to take care for those 100 days, which is very important, which is the golden period to heal that wound for that patient. Thank you, sir. Thank you once again. So we will now, yes, sir. So we'll be shifting to the hands-on workshop. I once again would like to introduce Ms. Sujata, who has been kind enough to come from a different city for Dr. Sunil to demonstrate certain things on her. Over to you, sir. Both will work together?
ಎಲ್ಲಿದೆಮ್ಮ ನಿಮಗೆ ಇಲ್ಲಲ್ಲ ಓಕೆ ಫೈನ್ ಯಾ ನೋ ಐ just see if you are having this with you, this is a nail clipper, and I think he is going to focus on this area for the callus. What normally you used to do it with the blade, you are not ready to accept it because you don't want to go with that sharpness. Sometimes you go deep, though you want to make minimum damage in that area. Trimming is the procedure what we do it, but with this we could see that if you imagine this is a foot and there is a callus somewhere, just keep it parallel, abutting to that entire callus, and initially just create a few clicks on that so that it raises up. It's a very simple technique. Keep abutting to the entire plantar skin around the callus and keep on giving approximate the edges so that the callus raises up, and then keep on shaving the callus with this. There will be no blood, nothing will be there. I will show you this. Can I have one chair so that I can be comfortable? Dealing, oh, that will be too high. One sec. Okay, chalo. No, that is okay. Can you focus here? Is it a nail new? Is it comfortable? Yeah, excellent. Yeah. Pawan, she is your patient?
Yep.
Why she is with such a nail?
She had not come for a long time. That's why we called her here.
Can you focus it? Yeah. It's there? Yeah. See, I think we are not happy with the nails also. Yeah. I am just abutting to the callus. Okay? Okay. So once you abut to the callus, try to approximate the edges. Don't cut it, just approximate so that the callus gets prominent. Once the callus gets prominent, you can just trim it off. I have done it. There is no bleeding, nothing. This you can do it. Don't go with the blade. This is available. This trimming will prevent first ulcer to 80% of the patients. Let it be on this area. Even she has got callosity on this area, lateral border of the great toe. Similarly, you can place initially parallel abutting to the plantar skin and just approximate, feel for it, how far you can raise it up, and then Noidreli, Noidanama, Ilala, okay. Nodi. Okay, we have done this. Now the most important, you have got a file. Nail filer is there, you just file it, whatever once you have done the procedure, that takes out periphery, whatever the callus is there, it will just take it off. So this, I think, every physician can do this. You can get away with this blade, BP handle and all that. But this can definitely help you, and you have got a lot of confidence. I think the way once you start doing it, initially you are trying to hold it, don't cut it immediately, try to raise it up by approximating the things, and then just shave it off at the top of the head. 80% of the job is done. And now, the most important is nail. I don't know. She hasn't ಹೋಗಿದ್ದಿಲ್ವಾ ನೀವು ಫಾಲೋಅಪ್ಗೆ ಡಾಕ್ಟರ್ ಕಡೆ ಹೋಗ್ತಾ ಇದ್ದೀರಾ I don't accept such nails. Okay. When you want to go for a nail cutting, go in the center. This is what is my practice. You know, I just cut this nail in the center first. The center, one click, it cuts away. The entire thing comes out. Then you just go at the edge, take out the one piece, from this edge another piece. I think that solves then the result. The file is enough. So in the kit I have given everyone file. Nail filer is there in the kit. So I want you to see that every patient, she need not go for nail cutting, she can use the file once she go after bath, it is wet and it falls down as a powder. So it's very easy for you to work on this area. If there is an ulcer here or an ulcer here, as I showed you, as a physician, they can always plan for just above ankle TCC. Above ankle TCC, once you lay it, you can cut it off and make it 2 holes. Every day you dressing, again you close it. Fine. So I think we are already late. I think I will demonstrate TCC some other time. Anytime you just, we will have one more session only for TCC. I want every physician to practice this TCC. This TCC was earlier planned up to knee high, which is not required now as per the guidelines. Even if you plan just above the ankle, that hits the bone, and we can work on that. So I have shown you the evidences, forefoot, midfoot and hindfoot. Everything heals within 100 days, and put that whatever the cast, removable cast you have designed into the footwear, accommodative footwear. So he moves with that. Patient is ambulatory, and that is how it should go. Any questions?
Any questions? Any questions? We are sorry if we have overshot. The idea was
Done.
to talk about. The idea was to talk about basics and to show you some basic techniques.
Very
We are
glad
so
video.
sorry that we have not been able to show you what ECC application. That's one of the major things. But we would love to show you sometime around. We can work on that. But as we are running short of time now, we will give the stage over to Dr. Manohar.
Truly an absorbing session. A good round of applause for all the participants. And I must apologize for giving them only 1 hour.
Thank you.
I'm sorry for that, sir. Next time definitely we'll accommodate more
Sure,
time for our benefit. Thank you so much. I think we'll move on to the next presentation. Dr. Shanmugavelu is there. Yeah. So we had to make some modifications in the like how we make modifications in the treatment, patient-centric, like faculty-centric sometimes. When we have so many faculties, it is to be done. So instead of my talk, Dr. Shanmugavelu will speak on achieving glycemic symmetry with newer basal insulin. I request Dr. Amal Selvam, my friend, to chair. He's a physician from Bangalore, and Dr. Vijay, you can chair the session. Over to Dr. Shanmugavelu. It doesn't need any introduction. Considered a very, very busy man, but academically very active. We have been working together during the COVID times also with some studies. And he's a central RSSDI member. Over to you, sir, for the topic.
Respected chairpersons, senior doctors, my doctor friends, I feel extremely happy, honored and most privileged to have been invited to make a presentation on achieving glycemic symmetry with the newer basal insulin analogs, with a particular focus on evidence-based findings. And of course, I thank Dr. Manohar, Dr. Vijay Kumar and Dr. Suman for their kind invitation. Also thank Novo Nordisk for sponsoring this particular program. This is an industry-supported symposium. My disclosures, of course, I have no conflict of interest in relation to my presentation. What I'm going to do for another 17 minutes, achieving glycemic symmetry with newer basal insulin analog. So type 2 diabetes is a multi-causal, heterogeneous, progressive cardiorenal metabolic condition. The progressive nature is one of the major challenges. So getting the patients to goal become challenging as the disease advances. Multiple intervention will need to be used. The relentless progressive nature of type 2 diabetes results in an almost inevitable need for insulin supplementation as well as insulin intensification. Several trials have clearly shown the benefit of early intensive glycemic control with reference to diabetes-related complications. Intensive glycemic control has been associated with increased risk of hyperglycemia. Therefore, achieving proper glycemic control, at the same time preventing the risk of hyperglycemia, are regarded as a major measure for managing patient with the type 2 diabetes. So achieving glycemic symmetry, financial disclosures, let us take a case study. So 45-year-old male, engineer by occupation. He presented to the OPD with a family history of diabetes and a medical history of taking or receiving treatment for diabetes, hypertension, dyslipidemia. He presented with very high fasting, postprandial plasma glucose, lipid levels, as well as A1C level. So if you look into this particular treatment history, that is, patient has been on antihypertensive therapy, statins, as well as cardiac drugs were also prescribed in combination with oral antidiabetic drugs. However, his blood sugar remains uncontrolled. He has been currently on metformin 2 gram, glimepiride 2 mg BID, MPOC 25 mg once daily. The primary challenges identified: uncontrolled hyperglycemia, number one. Subject has a hectic work life and fears insulin initiation will affect work life due to hyperglycemic risk. So the risk of hypoglycemia, uncontrolled hyperglycemia number 2. So travels often and unsure about taking insulin on fixed time daily. So we need to have a formulation which can allow for flexibility with reference to timing of insulin administration. It is very difficult to take insulin at a particular time every day, same time every day. So what next in this patient? ADA 2022 recommends basal insulin at initiation. If already on GLP-1 receptor analog or GLP-1 receptor analog not appropriate or insulin preferred, and if A1C is not on target, add basal insulin. That is the recommendation of ADA. So factors to be considered before prescribing a basal insulin. So basal insulin, there are certain requirements for an ideal basal insulin. It should be efficacious, number 1, and should be associated with low glycemic variability. So the risk of hypoglycemia will also come down. And next thing is it should be available in a simpler delivery device, which is said to be user friendly, so it is acceptable by the patient. It will improve the adherence, compliance, as well as quality of life. Significant A1C reduction across clinical trials. So if you look into basal insulin analog, IDAG, insulin degludec, it is an ultra-fast, long-acting basal insulin analog. And if you look into the RCTs, as well as real-world evidence, therapy has been associated with significant reduction with reference to A1C level. So it is found to be effective with reference to A1C reduction. So initiating insulin therapy with IDAG in Asian patient with the type 2 diabetes inadequately controlled on OAD provides similar improvements in long-term glycemic control compared to other basal insulins like IGLAD, that is insulin glargine 100 IU as well as 300 IU, but at a significantly lower rate of overall confirmed hypoglycemic. So therapy has been associated with similar A1C reduction, but with reduced risk of hypoglycemia. That is very important, which in turn will improve the quality of life as well as patient adherence. And very important thing is it also minimizes the cost, resource utilization, and so on. Once stable glycemic control in insulin dosings are achieved. So factors to be considered. Number 1, efficacy, right? So consistently proven hypoglycemic benefit. Next is with reference to hypoglycemia. Hypoglycemia has been considered as a major barrier for initiating insulin therapy, major barrier for implementing intensive therapy, and one episode of hypoglycemia is associated with fear, anxiety. This may have a negative impact on quality of life, as well as this will definitely lead to discontinuation of the therapy. And very important thing is likely to increase the risk of diabetes-related complications. So if you look into the RCTs begin, switch 1 and 2, patients with type 1 as well as type 2, and devote. That is, this particular study was conducted in patients with the type 2 diabetes with multiple risk factors for atherosclerotic cardiovascular disease, as well as patients with the type 2 diabetes and atherosclerotic cardiovascular disease. Compared to IGLAD, IDAG established similar glycemic improvement, number 1, and non-inferior with reference to the reduction in the risk for cardiovascular endpoints. Next is you treat type 2 diabetic patient. That is the real world evidence. All these trials clearly shown that therapy has been associated with low rate of hypoglycemia. So degludec achieving target study. So this particular slide clearly shows patient with the type 2 diabetes on insulin glargine 100 IU as well as IDAG. The same study was also conducted in patient with the type 1 also. This particular slide clearly shows the association between glycemic control. So kindly focus your attention on x-axis, which shows A1C. Next is y-axis shows with reference to hypo events per 100 patient per patient year of exposure. So if you look into the association between glycemic control and the risk of hypoglycemia, patients treated with IGLAD, the curve shows more or less a concave. On the other hand, the curve in patients treated with IDAG, it assumes a convex. So there is a difference with reference to the risk of hypoglycemia as well as the achieving optimal levels of glycemic control. So one can achieve lower glycemic targets with the degludec versus glargine U100 before incurring an equivalent risk of hypoglycemia. Right. Next is devote. Major adverse cardiovascular events in patients with a very high risk for severe hypoglycemia. So this particular trial conducted in patients with the type 2 diabetes with established cardiovascular disease or patient with the type 2 diabetes with multiple risk factors, both IGLAR as well as IDAG achieve similar glycemic control, but with reduced risk of hyperglycemia, and both the drugs demonstrated non-inferiority with reference to MACE. Next is conclude study. RCT in insulin experienced participants, and kindly look into the hypoglycemic risk. So insulin degludec has shown a lower risk of hyperglycemia compared to glargine U300. So glargine 300IU and insulin degludec both belong to second generation of long-acting insulin analog, and to achieve steady state, it takes minimum of 2 to 3 days. Once a steady state is reached, the plasma insulin concentration of insulin remains relatively stable, flat, so the risk of hypoglycemia is very well less, and which gets translated into pharmacodynamic property, namely it minimizes the excessive glycemic excretion. That is the main reason behind reduced risk of hypoglycemia when you use both the second generation insulin analog. Next is confirmed study, data from real world. That is insulin degludec has shown a significantly low risk of hypoglycemia versus glargine U300 in evidence from real world practice. So patient with type 2 diabetes, insulin naive, inadequately controlled on OAD, when given insulin degludec as well as glargine 300IU, you can see the change in the rate of hypoglycemia demonstrated 30% lower rate of hypo with degludec, number one. Change in the proportion of patients experiencing more than one episode of hypo was significantly lower with degludec compared to glargine 300IU. So these results may suggest that there are potential economic benefits by reducing the risk of hypoglycemia. We can reduce the resource utilization, emergency hospital visits, and next important thing is it will have a negative impact on the socio-economic burden, socio-economic status also. So economic benefits with the use of degludec versus glargine U300IU. Next is glycemic variability. Degludec, it offers lower within day variability versus glargine U100IU. And if you look into the panel on the right side and panel on the left side, panel on the right side shows a consistent glucose lowering effect over a period of 24 hours. In contrast, patients treated with glargine, you can see the inconsistency with reference to glucose lowering effect. Thereby, it suggests lower within day variability with reference to degludec. This gets translated into low risk of hypoglycemia. That is very important. And this improves the predictability. So treating physician will feel confidence of putting the patient on particular dose because the fluctuations with reference to glucose levels are very minimal. Next is degludec, lower within day variability versus glargine 300IU. So even within the second generation basal insulin analog, IDEG offers lower within day variability. So again, this particular panel on the right side clearly shows consistent glucose lowering effect. In contrast, patients treated with glargine 300IU, you can see a U-shaped curve, that is maximum effects are seen in the early 6 hours and the late 6 hours with a dip in the middle. Next is degludec offers 4 times lower glycemic variability compared to insulin glargine U100 and U300IU. So patients treated with IDEG showed in the blue color and patients treated with IGLAR 100 and 300 in other color. You can see the consistent glucose lowering effect that is maintained in patients treated with IDEG compared to patients treated with glargine 300 as well as glargine 100IU. And this particular study, time in range with IDEG and IGLAR 100IU, IDEG showed superiority in terms of improving the time in range by 20.6 minutes in contrast to patient treated with IGLAR. Not only that, it also reduced the time below range by nearly 12.7 minutes in contrast to patient treated with IGLAR. So it reduces the time below range, thereby the risk of hypoglycemia. It improves the time in range, thereby it helps to achieve the targeted A1C level. In range, it is yet another study. You can see the inclusion criteria, a large-scale prospective multi-center continuous glucose monitoring based RCT. You can see the treatment period, starting from the screening period, run-in period, and patients were randomized to receive Glorgine 300 IU as well as IDEG, right? You can see the continuous glucose monitoring, that is parameters. Different parameters have been assessed. So patients treated with IGLAR, patients treated with insulin degludec, with reference to time in range, with reference to A1C, time above range, greater than 180 milligram, less than 250 milligram, that is level 1, time above range. It appears there is a difference numerically, but not statistically significant. So overall, degludec compared to Glorgine 100 IU, 300 IU, it improves time in range by 48.24 minutes and time above range by reduction in the time above range by 44.94 minutes and A1C improvement. Next is simplicity. So simple initiation and titration of degludec in patient with the type 2. Start in insulin naive patients administered alone or in combination with OAD, GLP-1 analog or bolus insulin. Recommended daily starting insulin dose is 10 units. You can up titrate depending upon the fasting plasma glucose and measure 3 days fasting plasma glucose. Average of the 3 or the lowest of the 3 can be taken for up titration, and titration adjusts dose once weekly based on the mean of 2 SMBG results. So switch from other basal insulin. When you have plans to switch from other basal insulins or other premix insulins, can we replace it with IDEG? Yes, of course. So unit to unit switch or 20% dose reduction to be considered when switching from a BID insulin. If a patient is already receiving once daily, either a premix insulin or once daily basal insulin, you can change the same unit dose. If the patient is receiving twice daily, you can reduce the total reduction dose by 20%. So flexible dose. So it is very difficult to stick on to the same time every day for taking insulin administration. There are occasions wherein this particular time of taking insulin may not be possible. So IDEG offers flexibility with reference to timing of insulin administration. So this particular study clearly shown that without compromising efficacy and safety, it has established improvement in glycemic control as well as the risk of hypoglycemia. And minimum of 8 hours between dosing is mandatory while giving the dosing of insulin degludec. So better adherence with degludec, this particular confirmed study, you see the Kaplan-Meier curve. The curve starts separating much earlier. So 27% lower risk of treatment discontinuation were demonstrated with degludec versus Glorgine. So Glorgine 300 IU and degludec achieved similar A1C reduction. And the risk of hypoglycemia, no difference at all statistically, but numerically different. But the rate of discontinuation was very high in patients treated with 300 IU Glorgine compared to patients treated with IDEG. So likelihood of discontinuation of basal insulin in patient with initiated treatment with the degludec or Glorgine 300. So lower dose and lesser SMBG monitoring. So lower dose, that is 10% lower dose with the degludec compared to Glorgine 100 IU and 23% lower dose with the degludec versus Glorgine 300 IU. And 2 strips less per week with the degludec with reference to monitoring. So this will definitely improve cost saving. So ultra long acting insulin analog, which can reduce the risk of hypoglycemia and other sources, may improve the economic status of the individual, and it is cost effective. So progression to good glycemic control in this patient, insulin dose, and you can see the baseline 1 month and 3 months, and the A1C and with reference to body weight. So patient uncontrolled on multiple OADs, initiating and optimizing insulin degludec. So we have the oral medications and initiation, IDEG 10 units with the dinner, titrated over 12 weeks, and results, end of 12 weeks, fasting came down, post-breakfast improved, A1C decreased from 8.8 to 7.4, and no episode of hypoglycemia. Thank you very much for your kind listening.
Thank you for a timely finishing the session. So it was a wonderful presentation on the basal insulin. One question I had, one patient, if there is any special situation where or something, where the patient is on degludec and there is severe hypoglycemia, will there be a prolonged hypoglycemia that is going to be there, or that may lead to a higher, prolonged hospitalization, more cost involved.
That is, you want to say whether the hypoglycemia
Patient is having some illness where there is total intake, food intake is less, and the patient is on degludec. Patient undergoes hypoglycemia.
Since it is a long-acting insulin.
Yes, sir.
The hypoglycemia will be also long-acting. That's what you want.
And no, the thing is, we normally put the patient on IV glucose, and we continuously monitor the patient, and we try to keep the glucose level between 90 to 140 maximum, and we stop it, giving intravenous, and we monitor the patient every hourly or every half an hourly, depending upon the risk involved.
Yeah, my question was compared to other basal insulin.
No, we have not come across.
We have not come across. Stay for a longer duration in the body.
We have not come across. I think Prasanna Kumar sir, he can highlight on this.
Thank you. Any more questions from the audience?
Actually, he has given an answer partially. With IDEC or whatever you want to call it as, the time below range improves. I think this is an indirect, your question is very valid. When you give glibenclamide versus glipizide, what would be the long-acting hypoglycemic effect? It is true there, because it's an insulin secretor. But with IDEG, there is what is called as, after 4 days, what goes in, what comes out. Steady state metabolism occurs with IDEG. And TIR, TBR, time below range, improves very significantly. That means chances of hypoglycemia come down even though you are given insulin. Why? The dose will be much lower, 40% less than other long-acting insulins. So you need not worry that much.
Yes, thank you, sir. Thank you for the answer. Any more questions from the audience? Yeah. I think no more questions. We'll move on to the next session.
Thank you,
Dr.
Well,
Shanmugavil, for your wonderful.
Photo with the photographer. Photo. So we'll move on to the next session now. A new chapter for improved cardiorenal outcomes in diabetes with CKD. I would like to invite the chairperson, Dr. Sudhindra. Dr. Amal can stay back as the chairperson. And the speaker is Dr. Rajanna Sridhar. Yeah. Next up. Just introduce the speaker.
So I welcome Dr. Rajanna Sridhar. So his education qualification is MBBS from Mysore Medical College, Internal Medicine from USA and Nephrology from USA. I welcome Doctor to give his talk.
Good afternoon. Thank you to the organizing committee of RSSDI and my dear friends. I just wanted to give a brief overview of about this new molecule, which is likely to, or which has been shown to be significantly effective in preventing cardio-renal episodes in patients with diabetes type 2 and CKD. So we know that CKD is progression of CKD is driven by hemodynamic factors, metabolic factors, and inflammation and fibrosis. And this leads to tubulointerstitial damage and fibrosis, glomerulosclerosis, mesangial expansion, so on and so forth. All these processes lead to the progression of chronic kidney disease. Now, until now, we have been focusing on controlling the hemodynamic factors with ACE inhibitors and ARBs, and more recently with SGLT2 inhibitors, and metabolic factors such as the glycemic control. You are experts in that, I don't have to say anything about that. However, still now there have been no treatments available targeting inflammation and fibrosis. So the triad of hemodynamic factors, metabolic factors, and inflammation and fibrosis, we have been focusing on two factors, two drivers of CKD progression, not the third driver. And that is where mineralocorticoid receptor antagonism comes into play. That is MRAs for heart and kidney protection. That is the title or the focus of my presentation today. Now, mineralocorticoid receptors belong to the superfamily of nuclear hormone receptors and are expressed in hearts, kidneys, vasculature, brain, gut, and myeloid cells. They are activated by ligands such as aldosterone and cortisol, and non-ligands such as RAC1 oxidative stress, etc. Now, we all know that the MR expression controls fluid and electrolyte and hemodynamic homeostasis. However, more recently, the role of mineralocorticoid receptor in inflammation and fibrosis has been better understood since the cloning of the gene in 1987. So now, kidney and heart diseases, that is CKD and coronary artery disease or ischemic heart disease, and diabetes are associated with MR overactivation through multiple mechanisms, including activation by ligands such as aldosterone and cortisol, and non-ligands like RAC1, oxidative stress, etc. that I just mentioned. And in addition, there is also increased MR expression. So by various mechanisms, there has been increased activity of the mineralocorticoid receptor, and since we understood that it is involved in inflammation and fibrosis, so this potentially plays a huge role in the progression of kidney disease and heart disease. So this is the history of the mineralocorticoids in health and disease. In 1938, the cholinergic and sodium retaining effect of DOCA was established. In 1960, FDA approved spironolactone. So it took 4 decades, that is 1999, Rawls study to establish spironolactone as a life-saving drug in patients with heart failure. And subsequently, the emphasis trial in 2011 established eplerenone also as a life-saving drug in heart failure patients with reduced ejection fraction. Now, in kidney also, MRAs have been shown to decrease proteinuria in patients with CKD. However, the problem with MRAs has been the risk of hyperkalemia when used in patients with CKD. So, although spironolactone and eplerenone decrease cardiovascular morbidity and mortality in patients with CHF and reduced ejection fraction, and subsequently in the TopCat study even with preserved ejection fraction, and spironolactone has been shown to improve endothelial dysfunction and increase nitric oxide bioavailability. The problem has been that in patients with CKD and type 2 diabetes, MRAs have not been used in a significant proportion because of the risk of hyperkalemia and anti-androgenic adverse effects such as gynecomastia and increase in cortisol and HbA1c levels. So therefore, although we know it is effective, or rather it is involved in the progression of kidney disease and heart disease, we have not been able to use the molecule because of its adverse events. So there enters finerenone. Finerenone is a novel, non-steroidal, highly selective mineralocorticoid receptor antagonist. Novel, I say, because this molecule was taken out by going through more than a lakh chemicals and subsequently refined to make it available or used in the human, in man. And it is non-steroidal, therefore the side effects, anti-androgenic side effects are going to be absent. And it is highly selective because of its unique structure, and it is an antagonist which is a bulky molecule with a short half-life. Therefore, the hyperkalemia effect is minimized because of its short duration of action. It has a unique structure that results in selective and potent interaction with the mineralocorticoid receptor and the regulation of its gene expression. And more importantly, its anti-fibrotic and anti-inflammatory effects have been well established in animal models of heart disease and kidney disease. So it meets all the criteria to be a very useful drug in disease. So in phase 2 studies of this molecule finerenone, in patients with chronic heart failure and mild to moderate kidney disease, finerenone, or it was called as BAY948862 at that time, was at least as effective as spironolactone in decreasing biomarkers of hemodynamic stress in patients with heart failure and CKD. But more importantly, there was a lower incidence of adverse events of hyperkalemia and worsening renal function that was seen with spironolactone. In another phase 2 study, remember these are phase 2 studies with 823 patients. It's a very, very large phase 2 study. Patients with CKD, albuminuria, type 2 diabetes, there was a dose-dependent reduction in proteinuria, and these patients were already on ACE inhibitors. So on top of ACE inhibitors in this phase 2 study, finerenone caused a significant dose-dependent reduction in proteinuria. Now, because of this impressive phase 2 trial data, large phase 3 programs were undertaken. And there were 2 studies mainly, Fidelio DKD and Figaro DKD. These were complementary studies in that they had similar design with minor changes in the inclusion criteria. And because of similar design, they actually were able to do a patient-specific pooled analysis of these 2 trials. Now, the Fidelio DKD, the primary endpoint was a kidney composite endpoint of less than or greater than 40% decline in GFR, kidney failure, or renal death. And in the Figaro DKD, the primary composite endpoint was a cardiac endpoint. And the secondary endpoints for these studies were converse. In Fidelio DKD, cardiac endpoint was secondary, and in Figaro DKD, kidney endpoint was secondary. And there were other secondary endpoints such as all-cause mortality, hospitalization, change in urine albumin creatinine ratio, and onset of kidney failure or 57% decline in GFR. This is a very kind of a minute point, I would say. 57% decline in GFR equals doubling of creatinine. Most earlier studies took doubling of creatinine, dialysis, or transplantation as composite kidney endpoints. Therefore, this actually is a better endpoint, but 40% reduction was actually mandated by FDA, and therefore they took 40% as the primary endpoint. So there were other exploratory endpoints as well. So the design I told you was similar in both studies. It was a randomized, double-blind, event-driven. That means how many events occurred, then the study would be stopped. Placebo-controlled phase 3 trials. 13,000, nearly 14,000 patients were run into the study. The most important thing is optimal ACE or ARB therapy was established. That means 99.8% of these patients were already on ACE inhibitor and ARBs, and that was optimized in the run-in period. Then subsequently, after a 2-week screening period, 5,734 patients were randomized in Fidelio DKD, either to finerenone 10 milligrams if the GFR was less than 60, or 20 milligrams if the GFR was more than 60, or placebo. And they were monitored every 4 months and until the number of events accumulated in the study. Now, this is the primary kidney-specific endpoint of the Fidelio DKD study. On top of maximum tolerated RAS therapy, finerenone significantly decreased primary kidney outcome by 18% on top of RAS therapy. And I did not mention, for the lack of time, These patients had an HbA1c of 7.6% at inclusion and blood pressure of 136/82. That means extremely well controlled type 2 diabetic patients with CKD. On top of RAS therapy, there was additional decline in the kidney endpoint by an absolute risk reduction of 3.4%. That means the number needed to treat, I always tell the students, if there is one number you should take from a clinical trial, it is number needed to treat. It is nothing but 100 divided by absolute risk reduction. That means if you treated 29 patients for 3 years, you prevent one event, that is one kidney failure or a composite endpoint. So, by the way, 29 is a very good number. Okay. So, if you treated 29 patients for 3 years with finerenone, you prevented one patient having this endpoint. So, and also I would like to point out that for the first 12 months, there was really no change in this outcome. Only after 12 months, the lines started to separate. And also, you might say, at the end, they kind of came close together, but that is really irrelevant because the numbers followed at 48 months was really very small. So, it's not an important finding at all. Now, the secondary endpoint of cardiovascular outcome in Fidelio-DKD study also was significantly decreased by 14%, and here the number needed to treat was 42. Now, the secondary endpoint of urine albumin creatinine ratio decreased by 31% at 4 months compared to placebo. So, in patients with maximally tolerated RAS therapy, there was a further decline in proteinuria when finerenone was given. I told you that MRAs are not used in patients with CKD because of the risk of hyperkalemia. If you look at AMBER CKD study, which included patients between GFR of 25 and 45, and they were given ACRB plus spironolactone, 23% of patients had hyperkalemia. In the Fidelio-DKD study, 2.3% had hyperkalemia. I must say that Fidelio-DKD included patients between GFR of 25 to 75. So it is not apples to apple comparison, but still there was a significantly less number of patients with hyperkalemia when finerenone was used. Now, Figaro-DKD, I told you here the cardiac endpoint was the primary endpoint and kidney was the secondary endpoint. So what did we find here? Again, there was a significant reduction of 13% cardiac outcome on top of maximal RAS blockade, and number needed to treat is 47. You might say, oh, this is not so good as Fidelio-DKD, but please remember, Figaro-DKD inclusion criteria included patients between GFR of 25 to 90. That means these were less sicker patients in the kidney, from the kidney perspective. So therefore, 47 is also a very good number. So in Figaro-DKD, the primary endpoint was decreased by 13%, secondary endpoint was decreased by about 13% when the GFR was 40% decline, and 23% when the GFR decline was 57%. That is, doubling of creatinine again decreased by 23% in this study. So I just want to put this in perspective. Renal study, losartan was good, 1,500 patients, phase 3 study. IDNT 1,700, Credence 4,400, EMPA 6,600. But Fidelio and Figaro together had 13,000 patients, largest CKD trial to date with any drug. So when this 13,000 patients were analyzed in the patient level pooled analysis, there was 14% reduction in the cardiovascular morbidity and mortality, 23% reduction in kidney mortality or kidney disease progression. So therefore, without doubt, one can say that finerenone is an effective treatment option for cardiovascular and kidney protection in patients with CKD and type 2 diabetes. So based on these phenomenal data, all organizations, professional organizations in the world have now recommended finerenone. This is the latest guideline from KDIGO. SGLT2 inhibitor obviously is a first-line therapy in patients with diabetes, GLP-1 RAs for additional glycemic control, and finerenone if the patient still had residual proteinuria. It is not just KDIGO, American Heart Association, American Diabetic Association, AACE, so on and so forth. Everybody has given a level A recommendation for the use of finerenone in patients with type 2 diabetes and CKD. And there are additional studies that are ongoing when in patients with heart failure or non-diabetic CKD, whether it is going to be useful, or in combination with empagliflozin in confidence trial, or in patients with kidney disease, children with kidney disease. Therefore, a lot of indications are going to be, whether it is useful or not will be known in the very near future. But to date, type 2 diabetes with CKD, finerenone is definitely a drug that is going to decrease cardiac outcomes and kidney outcomes. So it was approved by DCGI in 2022 April, and it has been available in India since August of this year. So the take-home message from my brief presentation is that finerenone, a non-steroidal MRA, further improves cardiovascular morbidity and kidney outcomes in patients on top of standard therapy in patients with type 2 diabetes and CKD. You must consider finerenone if there is residual albuminuria and normal serum potassium on patients who are already receiving first-line therapies, and renoprotective effects. I did not provide the data, but I think I would like to emphasize that the renoprotective effects of SGLT2 inhibitors and finerenone are complementary, independent, additive, and makes the treatment combination more tolerable. So with this, I'm going to end my presentation. If there are any questions, I would like to answer.
So that was a wonderful talk, sir. So my first question is, do you experience any hypotension with finerenone?
No. The decrease in blood pressure with finerenone is very minimal, 2 millimeters on average in these studies. So you do not get hypotension.
Okay. And we physicians, normally we see our diabetic patients with stage 1, stage 2 of CKD. Which stage we are supposed to use this?
Like I said, in the studies, the inclusion criteria included, microalbuminuria, or rather, moderately increased albuminuria or severely, that is 30 or greater, GFR of 25 to 90. That means basically everybody. If the patient has microalbuminuria, you can use finerenone, provided you're already using ACE or ARB and their potassium is normal. And on top of that, of course, SGLT2 inhibitor is also indicated. On top of that, if there is residual albuminuria, you should add finerenone. I would say the way the CKD is driven by fibrosis, in most CKD there is fibrosis. So I would guess that earlier the better. Yeah.
Any questions?
These patients are already on ACARB.
Yes.
On the top of that, you are adding this drug.
Yes.
Can it not worsen hyperkalemia?
No, not at all, sir. That is why this large 13,000 patient studies did not reveal significant hyperkalemia at all. In fact, there were 0 deaths among 13,000 patients due to hyperkalemia. So hyperkalemia is a significantly less problem with this molecule compared to spironolactone or eplerenone. It is believed to be due to the short-acting nature of the drug. The hyperkalemia is a cumulative effect. Remember, potassium is excreted continuously, and spironolactone or eplerenone, by the longer half-life, the reaccumulation of potassium or reabsorption of potassium is going to be high, and with this molecule we don't see that.
Is it not necessary to monitor potassium in these people then?
Yes, like I said, initial potassium should be normal, that is less than 5. 1 month later, you should monitor potassium. If the potassium is less than 5, you can increase the dose from 10 to 20 milligram if you have started at 10. If the potassium is increased, let us say if the potassium went up to 5.6, then you can actually, normally we see potassium 5.6, we don't even get concerned. But if the potassium is high, you can decrease the dose. More importantly, in these studies, the patients were not advised potassium restriction at all. And most of our patients, we advise potassium restriction. So you can take other measures to keep the potassium low, such as low potassium diet, and restart the finerenone molecule at a lower dose, maybe alternate days. And if the potassium is well maintained, you can continue the treatment. So 1 month after starting the drug, you should monitor the potassium.
Low dose means 10 milligrams.
10 milligrams and 20 milligrams are the only two formulations currently available. So if the potassium, let's say if the potassium is 5.8, obviously you should withhold the drug, put the patient on a low potassium diet, maybe repeat the potassium after a couple of, again, see, this is not a, because the involvement is fibrosis, it is not an emergency thing. If you withhold for 1 month, nothing probably is likely to happen. Yeah. Any other questions? Thank you.
Thank you, sir, for a good presentation there. And now it's my privilege to move on to the next topic. My teacher, Dr. Prasanna Kumar, KM, is the speaker, and the topic is on emerging therapies in the management of diabetes. To chair this session, my good friend and colleague, Dr. Pankaj Singhai, senior consultant from Manipal Hospital, and Dr. Neelkant Kote, to take the chair. Over to the chairpersons.
Hello, check. Good evening, one and all present here. It's my privilege to introduce the speaker for the day, Dr. K.M. Prasanna Kumar. He's an endocrinologist from the Center of Diabetes and Endocrine Care, Bangalore. He was a former senior professor and head of the department in the endocrinology at MS Ramaiah Medical College, Bangalore. He was also the past president of KRSSDI. Between 2007 to 2009. He was a past president of Endocrine Society of India between 2008 to 2009. Past president Indian Thyroid Society between 2018 to 2020. During his tenure, he has handled various posts, including being the director of Primer, chairman of Changing Diabetes in Children, India, and he has got various publications, both national and international publications in the scientific journals, measuring up to 150, and he's up to so far he's presented about 20 orations at various conferences and seminars. Thank you. We welcome you, sir.
Thank you, chairpersons. Good evening, everybody, for the introduction. And at the end of the day, to have so many people is something rare after this COVID situation. That speaks volumes of the organizing capacity, and I would like to thank Manohar and Dr. Shekhar, who is the patron of this KRSSDI. And I think now the revival has come, and more people have come, and it shows the good scientific progress. I will restrict my talk to 17 minutes so that you have questions. Emerging therapies in management of diabetes. There are so many emerging therapies. I don't want to go into details of what is available 2022, what is likely to come 2023. I'm not talking of 2035. Some of us may not be here also. So let us talk about next 2 years. So these are the ones, imiglamine, which is a mitochondrial re-energizer, and lobeglitazone, PPAR, alpha and gamma agonist available. And of course, semaglutide is already available oral. You are likely to see next year Wegovy, that is injectable semaglutide. So much so, people say don't take it without doctor's prescription. They are not able to supply that. Celebrities, everybody says, I lost so much of weight. Of course, tirzepatide, which is another molecule with a GLP-1 and GIP receptor analog. A great thing, you will see the data. And of course, Icodec, we heard about the Degludec, and Icodec is once a week injection. Everybody asks, should I take daily, why not weekly? This is 2022 ADA guidelines. Last week, Diabetes Care had 2023 guidelines. December end it has come, January issue. All these things show that when you choose a drug, whatever it is, that just like Dr. Sridhar Rajana said, that look at atherosclerotic as well as high risk for cardiac disease and renal disease, choose the drug. Then you choose drug which causes minimal hypoglycemia, and also drugs which are cost. Today, everything has become generic. The what is available except semaglutide has become affordable at all. prioritize the use of organ protective medication. It's not that a drug reduces blood sugar, does it protect kidney, heart, now we are talking of liver also. So these are the mechanism, as all of you have seen, the octet, whether it is the pancreas, whether intestine, whether it is fat, kidney, muscle, brain and liver, and you have drugs, because by the time you diagnose 70% or 50% of beta cells are already gone. And various mechanisms, I said not one, there are more than that, even 10 or 11 as such. As you can see, the pancreatic beta cells, whether beta cell function reduced, beta cell mass also reduced, then incretin effect, then alpha cell defect, then you see the adipose tissue, muscle, liver, brain, and also you have got on the colon, then you have got immune dysregulation. Just now you heard about CKD, how the inflammation, then decreasing amylin, as well as small intestinal absorption, and of course kidney SGLT2 inhibitors. I think in the next future we may have many more mechanisms. For every mechanism there will be a new drug that is available. What is imiglamine? What is so great about it? It's a first-in-class molecule of glimins recognized by WHO for type 2 diabetes. Research was started in 2007, got approval 2021, 14 years. It's not a chance one. Its method of action, which is mainly on impaired glucose uptake by muscle tissue, acts on the muscle, reduces the resistance, excessive hepatic glucose genesis reduced just like metformin, and also increase beta cell apoptosis. This is how. So it stops the apoptosis of the beta cell. This is how it works on these mechanisms, and it has been done in various trials, all put together 1,555 studies, and it's called as times 1, 2 and 3 phase 3 randomized clinical trials. This is what the imiglamine molecule, it just looks like a metformin. So molecule wise metformin, certain actions are like metformin, but the mechanism of action is mainly on the mitochondria. That's why it is called mitochondria flexibility, mainly RAS production, apoptosis is reduced, ATP generation is increased, because of which the energy gradient across the membrane of the respiratory transport mechanism to mediate insulin secretion. It does not cause hypoglycemia like sulfonylurea because it is only dependent on the glucose. If the blood sugar is high, it works, otherwise it does not work. I don't want to go into all the details, for it is sufficient. What is the dose? It is available in 500 mg and 1000 mg. The dose is twice a day. You start with 500 mg twice a day, then you can go up to 1000 milligram twice a day, and it can be used as standalone or it can be addition to metformin. Of course, all over the world, every guideline says metformin is the first drug, ADA guidelines, WHO, everything, except the European Society of Cardiology said SGLT2 can be used ahead of the metformin. We don't agree because there are no diabetologists, they are all cardiologists. It's okay, it's all right. Whatever they can say, it is good enough. I and you have to treat that. It's the first in class unique action, improves C2 redox factor, modulates the blood glucose excursion, beta cell function, hepatic glucogenesis and uptake. And there are many, I don't want to go into detail. There are studies to show monotherapy. It reduces about 0.6 to 0.7 A1C reduction when added to metformin. So you can add to metformin, you can add to other drug, you can even add to insulin also. So all these cases are there. So if the patient does not tolerate metformin, you can start it, or after metformin you can use. And you can use with metformin, gliptin, then imiglamine, or metformin, gliptin, SGLT2, and then this, or metformin, insulin, all. Every data shows more than 3 drugs is useful, but everybody in this hall is using 5 drugs also, I know that. What you learn in dark, you don't practice in the light tomorrow morning, 10 o'clock in the morning. So talking is different thing, practice is different, because there are patients who say, doctor, one more drug, let me say, all right, take. To his good luck or your bad luck, he comes back, look, 6.8. You said after 3 drugs, no, now I am on 4 drugs. And has anybody started insulin without adding sulfonylurea? Because sulfonylurea is the most potent drug. So that's why, just like what is resistant hypertension, more than 3 drugs, one of them should be diuretic. Same thing applies to sulfonylurea. We have lobeglitazone. What is it? It's another glitazone, like Rosi and Pio. Then what is great about it? It's a novel glitazide where approved in Korea. The previous imiglamine also is not approved by FDA. That is approved in Japan, Korea and India as such. And this is the structure. Only thing is, Unlike the Pio and Rossi, lebeglitazone is a dual agonist against PPAR alpha as well as gamma. Everything else is just like that, except probably that lebeglitazone is less than 1% excreted in the kidney, and majority of them is through the liver. And the dose is smaller. It is 0.5 milligrams once a day. Whereas you see pioglitazone is 15 once a day, 30 once a day, not in India. Some countries they use 45. Of course, rosiglitazone is already gone. So it was not inferior to pioglitazone. So lebeglitazone, just like pioglitazone, 0.5 milligram, and it is safe, effective both alpha and gamma. Now the problem is obesity. As all of us know, 65% of the patients are obese. I want to have a study, India, Pakistan, Thailand, and other countries, and they said the BMI should be less than 25. Out of my 100 patients, I don't have 20 patients whose BMI is less than 25. I'm sure that is true in this hall also. So how do you expect our patients to be that? So that is why we want drugs which can reduce obesity, and this is what is a peptide, which is a dual GLP-1 and GIP receptor agonist as such. And there are so many, surpass 1, 2, 3, 4, 5, 6 studies. 13,000 patients have been done, and the participant even starting at about 8% experience 2.5% reduction, which is great. But what is so great about it? Weight loss, 52 weeks, 10 kg. If we can lose 10 kg, everybody here, we will live another 5 to 8 years longer. Even with SGLT2 inhibitors, studies have shown 2 to 3 kg. But today somebody came and said, my wife, everybody is telling, you have lost too much of weight. How much? 8 kg. Why are you worried? Sir, if I lose weight, nobody is afraid. Why should anybody be afraid? What are you? Sir, I am Panchayati member. If I stand for election, nobody will be afraid. The problem today is your patients say, no, my wife is telling your face is all dried up. You should be fat, okay, then you are good. This is the problem in our country. Anybody loses weight, maybe he is having cancer, heart attack, and he is not telling. So patients are worried if they lose 5 kg. So though we are happy, everybody is happy, patient is not happy, that is the problem. And I think if it comes not only that, the cholesterol, if you see, at 52 weeks, 6% reduction of cholesterol, triglyceride 22%, LDL 7.9, VLDL1, and HDL increased by 10.8. No drug will increase HDL. Look at this, this is fantastic. So if you lose 5 to 10 kg or 5 to 10% of your weight, I think your cardiovascular risk comes down greatly. Of course, I don't want to targeted therapy for adjunct, that is microbes we are using, all probiotics and all, this is something which will come. This is what I was telling you, results of it. Look at the SURPASS studies, what you will see here, step 2 key results. If you look at placebo, semaglutide and all, the reduction is great here. This is what you will see at 68 weeks. And these are all the studies with semaglutide, oral semaglutide is available now, and you can see a great weight loss in these patients 68 weeks, and on treatment you can see there is a great improvement. So this is what not only HbA1c, which reduced from 8 to 6.5. This is fantastic weight loss you will see, 1.5, 1.6 from 1 to 2.4 milligram. This is once a week injection. Thus, achieving A1C weight loss endpoint, if you look at it, this is something which is great. And look at placebo, semaglutide injection 1 milligram, 2.4 milligram. That's why Wegovy is right now we see it's a great one. The oral semaglutide, all of you have experienced. They thought probably it will make about 50 crores, 80 crores. They made 120 crores in India. Who thought anybody will buy 360 rupees tablet? I think you are mad. Anything 50 rupees, nobody will buy. And you have seen in 8 months that it is something like a blockbuster which nobody expected. And that's why you start with 3 milligram, go to 7 milligram, 14 milligram. We have now more than 6 months of experience. At least 40% of my patients have reached 14 milligram. Everybody will not reach 14 milligram. 40% is very good number what you see oral, and this is what you see how 6 to 8 core defects, whether brain, adipose tissue, liver, are all targeted when you use the GLP-1. This is what you will see semaglutide structure, and now we have got oral, and the most important thing is you should take it in empty stomach with 120 ml of water. Not very small, not well, half a cup of it. 24 clinical pharmacology trials, 9,543 patients, and these are all the trials going on in them. And this is what known as Pioneer 1, 2, 3, 4, 5, 6, 7, 8. I don't want to go into details compared to the monotherapy, Yampa, Sita, Lira, and renal and others one. You will see great reduction in the HbA1c as such, as high as 1.4, 1.6 you will see. And also, you will see A1c from baseline, it can go up to 2.3 also compared to the other sitagliptin and other drugs. So this is what the more than 5% weight loss in semaglutide compared to 10% in the tirzepatide molecule as such. This is the oral one, you will see superior reduction in A1c, cardiovascular safety, and this is what is established. So this is how you must take as soon as you get up in 120 ml of water. Don't have anything 30 minutes, don't take other minutes after the medicines, and first month start with 3 mg. Patient tolerates well, go to 7 mg, tolerates well, go to 14. Patients have already taken injectable, start straight away with 7 milligrams. And most of my patients are quite happy with the drug, as you see. This is what I told you, weight measurement, step 1, 2, 3, 4 trials. Some of them have achieved 15% of weight loss from the baseline, which is fantastic. And just like liraglutide, start with 0.25, go to 0.51, 1.7, maximum dose is 2.4 milligram over a period of 20 weeks, or you can say about 2.5 months, 3 months, you can achieve it. Beyond that, you get CV risk factors also comes down, and A1c comes down. This is the summary of semaglutide. 2.4 milligram injectable, not available in India, already approved by DCGA. You will get it next year, and this is the dose. Your safety profile is similar to other GLP-1 receptor agonist. And the last thing is once a week injection, icodec, and you can take it once a week, and by 3-4 days injections you will get a steady state. Hypoglycemia is much less, and as you can see, once you inject subcutaneous, goes to blood vessel, target tissue, it is slowly there, and you don't have to do daily blood sugar. Once a week, once to 8 days, like somebody in the previous talk telling, you can reduce the number of blood sugar tests. Each strip costs 20 rupees. So 3 times a day testing is more expensive than the insulin you get. To summarize this, we have lot of drugs in the pipeline next 10 years, but these are 4 or 5 which I think very, very important. Imiglimine, and you have got labetasol, and you have got tirzepatide, and icodec long-acting insulin, and semaglutide oral and injectable Wegovy. And patients and physicians prefer fewer insulin, so once a week will be useful. You can use it only in type 2, not approved by type 1, where it is very, very flexible. Patients have already reached a steady state. So last 3 months I am taking 20 units, 40 units, take like that. And it offers better convenience, better compliance, and available data shows they are quite safe. Thank you for your attention. If there are few questions, I'll be happy to answer.
Yeah, thank you, Dr. Prasanna Kumar, for a wonderful presentation and enlightening us about the newer generation, or probably I would say the emerging therapies in management of type 2 diabetes. And since the time of introduction of the newer generation anti-diabetics, we are not able to address the diabetes and its complication in a proper manner. So, what is the role of iminim in combination with the metformin in our population? Since we know the Japanese, they don't respond to the metformin well. I think probably they're not using at all. So that could be the reason most of the studies is done in the Japanese population. So what is your take? Probably we can consider the first line therapy in combination with metformin?
Actually, I didn't show it because of paucity of time. There is already Indian data of imiglimine with combination of metformin. And it showed over and above metformin, 0.6 to 0.7% reduction of HbA1c good. And also it showed that the safety profile is very good, and there is no problem in most of them didn't have. In fact, the gastrointestinal side effects are the only ones you see. It is slightly less than, because 15 to 20% of metformin they cannot tolerate, whereas imiglimine they tolerated better. The only indication, if they cannot tolerate metformin, then you can use imiglimine. Otherwise, start metformin, add 500 mg BD, and after 2 months, make it 1000 BD. The tolerability is good. This is about 700 patients, Indian studies.
Absolutely. So you rightly mentioned about the people, they don't want to see like they're reducing the weight. So like people are coming with a like significant loss of weight with the semaglutide. You know, you have seen like 5 to 10 kg
Yeah.
weight reduction. Probably in obesity, we have to treat in toto, you know, in terms of dealing the obstructive sleep apnea and obesity, diet control, and then newer generation oral GLP-1 receptor agonist. So some of the patient, despite of reaching the 14 milligram tolerating nicely, they're not reducing 1 kg weight also. So what could be the probably reason?
Very correct. There are 4 types of response. When you use GLP-1 analog, best response, they reduce weight also, reduce HbA1c. Second, weight comes down, HbA1c doesn't come down. Third, HbA1c comes down, weight doesn't come down. The worst is neither weight nor HbA1c. If somebody says, I have seen everybody has that, you are telling a lie. You are 100% correct. Let us say you give 3 months, doc, I don't find any improvement at all. I am spending 10,000 rupees per month. My HbA1c has not come down 0.5%. I have not lost 1 kg. So, every drug in this world has responder, non-responders. We don't know who is responder, who is not. And there are some patients who are on injectable, very happy, changed over to oral. Worse than that, we go back to injectable. There are patients. I still feel a lot of patients are doing very well with weekly dulaglutide. They don't want to go to oral tablet because it is more expensive. So, you have to individualize patient. You are not happy more than you. Your patient is not happy and spending so much of money. I think better to stop and say probably you are the one who is not. But we don't have a marker to say at the beginning who will respond, who will not respond.
Absolutely. Yeah.
Thank you.
So any questions or any comments from the audience? Yeah.
Sir, I should tell the diabetes population, doctor population here, one of the major reasons why I became a diabetologist is you.
Thank you.
Talia bajaiye. Now, the question is, how do you stay so reliable, so dependable, and so inspiring?
I think...
I need an answer.
Yeah, exactly. Wisdom comes from experience, right? And experience comes from mistakes. So nobody is a born speaker or anything when you start your career. And somebody comes and says your talk is not good, honest and all. Over a period of time, it becomes easy. Next thing is, when you talk from your heart, you don't need any PPT to kill. If you have an experience, knowledge, it's easy. If somebody gives us, these are the slides, then turn layer 20 minutes and then say anything like that. So turn to see that what's your experience and have you understood the concept. If you yourself has not understood, you will not be able to convince others. Sometimes they come, I think today your talk, you yourself are not convinced that it's not good. Somebody honestly gives that, you know your heart and mind is not in that topic, you just come and give some talk. So all of us can come to some extent, but everybody cannot be a born speaker. There are some people I have seen in my lifetime, nobody can beat them. One of them is Bajaj. How many people in this have heard Bajaj? Outstanding speaker in my lifetime. And whether he speaks to a nurse, undergraduate, postgraduate, or Indian Institute of Science professors, he comes so prepared. I will target Shekhar. He will rip you apart in that meeting. So that, I mean, it's other way of doing it is no more. So I think all of us have to learn from others. If you are a good listener, others, how good he is speaking, and you can learn. Otherwise, come here, okay, my talk is over, I am rushing away, then we are not a great learner from that. Yes, Dr. Shetty.
Such a humorous talk from a serious looking person. I think, and that says all about Dr. Prasanna Kumar. And those so thought-provoking talk. I'm just wondering, do we really practice the right diabetes practice as on today? Because the hemoglobin you put it, it looks like a panacea for all our problems. Such a wonderful drug. After that you came with other drugs as well. Now my question is, among these drugs, which one do you prefer? Number two, secondly, insulin is thrown away into the gutters. Number three, were we practicing all wrong medicine till today? These are my questions.
Thank you. 40 years back it was easy. You had only 2 drugs, metformin, sulfonylurea. You didn't have anything. Today you are spoiled by choice. You have got after metformin 10 drugs. So the question comes, which is second? And EAST, all guidelines says patient choice. First you say, this is the drug, it's a tablet, it's an injection, this is the doc. Don't tell me that I can't afford. So a patient choice is always the best. And tell them this is the cost, these are the side effects and all that, then he will choose. I don't say any drug is panacea, including today metformin is still the best drug. After that what? You have got 20 choices, including bariatric surgery and all. I think in every patient, when the patient is sitting in front of you, in that 30 seconds, your 40 years of knowledge, studies, whatever it is, in 30 seconds you have to decide, is this the best drug for him? And tell the patient this is what it is, and patient accepts it, fine. He comes back after 3. Every day I get 2 calls, you gave me this drug, I had nausea, vomiting, tablet is coming in this stool as it is. What do you say first thing? Yes, it could be side effect, please stop. If you try to insist, no, it's very good drug, continue, he'll never come back to you. So I think patient acceptance is the best what you see, and there is nothing like one prescription for all. Let us say you take 10 consecutive patients, it will be 10 different things. Why in this patient in 30 seconds you have to say he is overweight after metformin SGLT2. He is not overweight, I will use gliptin. I think this patient has so much of weight, I think he can afford, I will say GLP-1. I think I use all the drugs, but ultimately when I look at the patient, I know him 5 years, 10 years, I offer him, tell him these are the side effect, cost of it, let him decide. Most of the patients say, doc, don't worry about it, I don't bother the cost, give me the best. Then you have got a choice, whatever you think best. Whether HbA1c reduction, cardiovascular safety, renal safety, taking all this today, I got few drugs. Certain drugs might not have gone CVOT trial. You don't know they are cardiovascular safe, renal safe. Probably in such patient, you don't use those drugs. Very good. One thing is, guidelines are only guidelines. Guidelines are not a law. Guidelines are for MBBS, not for you. Dr. Govindappa with 50 years of experience does not need any guidelines. So, guidelines is only, and even in guidelines, if you look at it, what is it? Hardly 30% have level A evidence. Do you have level A evidence? No. 60% do not have level A evidence. Guidelines is you have to walk like this. What happens if you go, you can go in this way. I think it is better you walk that way. That's all what it guide. Don't think that, and who is it that is making guidelines? He is not any Brahaspati or anything. It's not Bible, Bhagavad Gita or Quran or anything. And many times people don't believe the guidelines. Guidelines are only a guidelines, not a law. Your wisdom, I think with 40 years of experience, better. You don't have to go a guideline, follow page 1, 2, 3, 4. I don't think it is necessary. It's a first MBBS student, sir, I don't have experience, so much knowledge. Then a book is taken, follow the guidelines. That's all. Beyond that, don't give so much of importance to guidelines. Yes, sir.
What is your experience with these drugs?
If you ask me, I have just started them. I don't think I can say today after 1 month. Some of them have been introduced 1 month. You can't say my experience of 5 patients, 1 month, what happened. Whereas semaglutide, I have more than 50 patients. It is good, as you said. It works in some, doesn't work in others. Other 2 drugs, icodec and trizepamide, I don't have any experience. Only semaglutide I have. And lopeglitol, I don't have the experience. I have just started. So also imedium. See, best thing is to use 5 patients. Wait 3 months. He comes back, you are happy, patient happy. Unless you have own experience, whatever you read, I don't think you can make any conclusion out of it. And why today metformin and others you have used gliptin, SGLT2, your own experience. I think Dr. Mano is getting restless. Okay, thank you.
Sir, sitting here on the podium, being a part of the session as a chairperson, we thoroughly enjoyed your session, and of course, we completely agree with you that orator skill is a very important skill for a doctor, for patient communication, as well as for having wisdom. It is important to communicate to the juniors and fellow colleagues. We highly appreciate your session. Thank you very much.
And just before we conclude, just congratulate Dr. Manohar, Dr. Shekhar and Dr. Vijay, Dr. Sanjeev Rao for having the fantastic KRSSDI feast over here. Thank you.
We'll take a clap.
Sorry for the delay, but sometimes it's out of hands. We'll move on to the next session, co-formulation insulin in Indian realities, from published evidence to practical experience. To speak on this, my good friend Dr. Faraz Farishta, endocrinologist from Hyderabad, and to chair, I would call upon Dr. Vishwanath K. and Vishnupriya Reddy onto the dais, and over to Faraz, and I'm always the villain, and I will request you to be crisper than usual.
Thank you so much, Dr. Manohar and the entire KRSSDI team for inviting me for this. And I see a lot of journal, and I would say very enlightened and energized faces. Thank you to Dr. Prasanna Kumar sir. I think I just missed him. But he has done the right dose of caffeine in all of us. We all needed that. So I think I will continue with that and boost my presentation. So these studies, we've always seen that, we all know that higher the intake of carbohydrate, the more risk you have of developing diabetes. And these are the two studies which have already shown. The pure study, again, which said more the intake of carbohydrate, the more increased risk you have. And the starch study, which was done across pan-India. Most important thing which I would like to highlight here is, unlike our Western population, where we always feel that once we get down the A1C, there the role of PPG becomes more important, and there it becomes very prima facie that we control the PPG. But in Indian populations, I would say, हम लोग हैं अलग अलग, हम लोग हैं जुदा जुदा. That is how it is. We are different. In every ways we are different. We carry a central obesity. So it is very important for all of us to control the fasting. It is very important to control the postprandial as well. So there are key issues when we practice in India. Primarily, first, across all the zones, if you will see, first is the high intake of carbohydrates, the central obesity, delayed insulin therapy. I think this is where it is hitting us very, very hard, that the inertia to start insulin therapy. My senior colleague was saying two drugs, 3 drugs, 4 drugs, but again, it's very easier said than done. Probably, I think it's time that we all are learning from it. And need for simple and convenience. I think whenever we talk insulinization for our patient, the first thing we see is the startle on their face or their attenders. I mean, we've seen that a number of times. Even if the patient is not astonished, the attenders start getting that fear. So we need something very simple. At the same time, we need something which will help us to control both the fasting and the postprandial. So which insulin regimen can address this? So before going to that, how many of us have watched the T20 World Cup this year? Oh, lovely. So we have a lot of hands in the air. This tells us two important things here, that with a great batting lineup, you can win a match, but you will not be able to win the final, or you will not be able to win the tournament. Again, same thing. With a great bowling lineup, you could reach to the finals, but you will not be able to win the tournament. So what does it tell? It tells that the better team, which had more all-rounders, had batsman and bowler and an all-rounder, won the tournament. So in our journey, in our war of getting control of diabetes, we will need a player. We will need somebody who would not only help us to control fasting, but postprandial as well. So when we talk about insulinization, I think a decade before where we all started this, fix the fasting first, where the concept came of basal insulin. Does your basal insulin answer these questions? Is it suitable for your high carb diet? Does it has any effect on PPG? Is it suitable when you are delaying insulin therapy, and whether it gives you once daily control? Your answer is no. So do we need something else? Yes, to answer this, we have the co-formulation. I think I do not need to speak much on that. It is a co-formulation, it is not a mixture. Remember that two drugs maintain their individual PK/PKD profile. That is very, very important, and that is the beauty of the drug. There is 70% of ultra long acting and 30% of the short acting. So what does the short acting does? The short acting helps you to control the postprandial rise, and the long acting helps you to control the fasting and moreover. So this is what is saying that once you give it once daily before a major meal, you control your postprandial spike and fasting. So this is a head-on study which was done once daily of insulin degludec aspart compared with the basal insulin glargine 100. You will see there was a better and superior A1C, 1.4% more reduction in A1C and similar fasting plasma glucose control, which is very, very important. And we all know that PPG rise itself has its own problems. It can cause endothelial damage. It can lead to all the increased risk of developing micro and macrovascular complications. So IDEC ASP is one drug which will help you to reduce your postprandial and fasting blood glucose. This is the SMART study. Again, this is a study which was done in India by my senior colleagues. Adults with diabetes where the participant received the IDEC ASP, the data was collected at 0, 3, 6 and 12 months. It was a multi-center, so it was spread well across the country, and the key inclusion criteria were the sugar control, fasting, postprandial and A1C. Here you will see the A1C reduction was around 1.8% in patients who were on insulin treated, and on oral anti-diabetic drugs, it gave you an A1C reduction of around 1.7%. So if you see the fasting plasma glucose reduction, it is around 50 milligrams. The postprandial again is the very important component, especially in a country like India. Here you will see it is 74 milligrams per deciliter. When you're starting a drug like this, one thing which will come to your mind, whether I am putting my patient at increased risk of hypoglycemia, the answer is no. So this study, you will see clearly said there was reduction in the number of hypoglycemic events before the start of the study and after 1 and 3 months. The main reason again being the less amount of dosage of insulin required to control the sugar levels. So insulin IDEC ASP is a very simple and convenient solution for all our diabetic patients. It can be given once daily. You can give it with the major meal. So you can give it for lunch, you can give it for dinner. It gives you a lot of flexibility, especially I would think a state like Bangalore, where more of the IT hub is there, where you do not have your patients maintaining that timing. I think we're doing that same in Hyderabad as well, nevertheless, but it gives you a lot of flexibility, 6 hours plus or 6 hours minus. That's a huge amount of flexibility you get with insulin. You can give it once daily. How do you monitor it? So if your fasting target sugar is above your target, you keep on adding 2 units every week. If it has reached the target, you continue the dose. If it is less than that or less than 100, you reduce the dose by 2 units. So where do you use the IDEC ASP in a diabetic patient? Use in patients where you are failing to achieve the A1C, where you want a good postprandial control, where there is increased risk of hypoglycemia, where there is a severe glycemic variability, and where you really want the flexibility in the timing. So to summarize, high carb and high PPG levels are the major problem of type 2 diabetes in our country. IDEC ASP once daily is a very simple solution for that. And we have a lot of analysis, we have real world evidences, data. I've been a part of 3 trials, even one trial which was done during the period of Ramzan as well, where once daily compared to the other premix insulin, it did show that the incidence of hypoglycemia was very less. So to end it, at the end of the year, I would still say we'll just continue with the World Diabetes Day theme of the IDF, diabetes, education to protect tomorrow. If we not do it today, then when? Thank you so much. And I'm sure, I don't see Manohar here, but I'm sure he's going to be very happy with this.
Thank you for the wonderful talk. In fact, even I was about to say that, right? It was crisp and you finished it very quickly. But, you know, in spite of so much of advancement, in spite of, you know, we having such good devices, still you will agree that there's so much of resistance to, you know, insulin. And, you know, with this flexibility, I think people are more accepting and able to take compared to earlier that half an hour. So,
one of the slide you said indication after adequate use of other insulin non-optimization, then you start, or can you start it as a first line? Because
now when we are talking,
that was the first indication you had put, that after other.
Yeah, see, that is why I said a decade before when we used to talk about insulin, I think all of us loved basal insulin. Yes. If I had to keep my hand here, and in my last more than a decade of journey of practice, if I have to talk. If I have to sleep well, my patient has to sleep well, it has to be basal insulin. You are not worried about hypoglycemia. But now we have learned that in that journey, we are missing that good glycemic memory.
Yes.
More and more inertia is there. So you start with basal, you intensify, it takes real time. So there you are putting your patient at lot of risk of developing micro and macrovascular complications. We do not want that. I think now the guidelines are saying that intensify the therapy, start it early. So maybe after metformin therapy, if your patient is having a higher A1C as the guideline says, 9% and above with osmotic symptoms, then definitely, because again that slide, if you will see the Moiner et al., where in Western population, the PPG control was important when A1C was reaching the target. But unlike in India where you want PPG and fasting to be under control. So if you want a once daily therapy, you're trying to look at the PPG, especially if you come to Hyderabad, we can't live without biryani. So, so what do you do about that postprandial rise? Similar in, I would say, Karnataka or anywhere in India. So when you're looking at both, you can start it initially with 10 units and then escalate the therapy.
So yesterday, I think in paper it came, Indians had ordered 2 biryanis every second. So that was the, I don't know, in types of India it had come.
Most
so I think all the more gas is needed.
I'm sure Hyderabad was topping the list.
Yeah, definitely. Thank you. Any comments? So Dr. Shekhar sir is here. We would love to listen to your comments.
About biryani and the postprandial rise.
He said IDAG is one indication, you know, when you eat a lot of biryani.
Please.
Anyway, we are a close-knit family now, not many around. My opinion about IDAG is it's probably the best. Just for the sake, not just pharmacokinetics and pharmacodynamics of the drug, put together my personal experience. I have been using Novo and I have been screaming at them for all the stupid information they come with pre-mixed fast-acting insulin and all that. But the paradigm shift happened when I had difficult to treat type 2 diabetics. I put them on IDAG, and now IDAG asp. Let me tell you, the so-called blood sugar control was pretty good, but much more important than was 2 things. The first one was hypoglycemic episodes. My patients on insulin were a little bit worried about hypoglycemia because you should go through hypoglycemia to know. Isn't it? That's what we tell. The second one was weight gain. With insulin therapy, you have no other choice but to tell your patient you'll put on weight. But with IDAG and probably IDAG asp, I have not had that. So I could probably talk about pharmacokinetics and dynamics, that it's steady state. You give 10 units, after 4 days, 10 units come out. So there is hardly any reliability component of, you know, glargine or whatever it is. The second one is weight gain, which everybody says is a done deal with insulin, has not occurred with IDAG. 5,000 rupees, please.
Absolutely, sir.
That is on the guy on the table, sir.
Sir, in fact, personally, when we, in my practice, what I saw, from basal bolus also, I was able to switch it to idagas with 2 doses. Instead of 4 shots, the person was taking only 2 shots. Then weight gain also was definitely much lesser, and she was very happy. Smooth control.
And then compliance improved significantly.
Compliance, she was so happy. 4 shots came to 2 shots, and that was possible with this.
Thank you, Dr. Farishda. It was excellent. As she said, very crisply you summarized the thing. Thank you once again.
Thank you.
Thanks. We'll just have a snap. So, penultimate talk. I don't know if I am growing old, finding it difficult to focus. Anyway, pioneering the upstream approach in diabetes management with oral semaglutide. I think we had some interesting pointers from Dr. Prasanna Kumar on this to speak. I would invite the chairpersons, Dr. Sanjay S. and Dr. Sridhar. For chairing the session, speaker is Dr. Jay Prakash, consultant endocrinologist from Kochi. Chairpersons to take the stage. Over to the speaker.
Yeah, I welcome Dr. Jay Prakash. So please continue.
Thank you. Thank you, respected chairpersons. Let me begin by thanking the Karnataka RSSDI for giving me the opportunity to talk, especially Dr. Vijay Kumar and Dr. Manohar. It was a well-attended conference, and it's glad to be back in the physical form. So I'll be talking on the diabesity and the role that semaglutide has played in terms of managing this. Actually, Dr. Prasanna Kumar's talk has made my talk much easier. He has covered most of the salient aspects. I will just add some icing to the cake that he has already served. The burden of diabesity is something which I think we are still underestimating. Again, the data which paved way for the diagnosis of obesity, bringing down the BMI to 23. Even with that, now again, Dr. Prasanna was telling about making it to less than 20. That's what the obesity starts with. But then, even with this data, there is an increased all-cause mortality. When a patient has diabetes and a BMI of more than 23, there is a 7 times increased mortality rate. So this kind of twin epidemic that we are facing is, there are various terms by which you describe this. Adiposity is one of those, where adipose tissue is at fault. Adipose tissue is actually now considered as the largest endocrine organ. There are so many chemicals coming out of it, which some of them are harmful, some of them are protective like adiponectin, but most of them are harmful, and most of this actually kind of drives the pathway of insulin resistance and cardiovascular risk and ultimately lead to diabetes. So the complications due to increased body weight, we know the cardiovascular complications, the micro and macrovascular complications, but the non-cardiovascular complications again go with that. Depression is commonly underestimated, obstructive sleep apnea, then the osteoarthritis, which is again debilitating. All these things are associated with obesity. So what you mean by this upstream approach is that when you reach the downstream, where already the complications of diabetes, the vascular complications and set in, there is a limited scope for our intervention. But when you travel upstream, when you try to catch the adiposity or adiposopathy and obesity, we will try to prevent these downstream complications. That's what we are trying to do. So this weight-centric management right from the beginning would help to not only to prevent the diabetes and the vascular complications, but it kind of disrupts the underlying mechanism so that people won't develop the complications. Weight loss as part of Diabetes Prevention Program or DPP trial is now 2 decades old. And it's shown that weight loss is one of the most effective measures by which you can prevent diabetes in somebody who was obese. So even a 5 kilogram of weight loss actually would help to prevent diabetes onset in people who are at risk for diabetes. What about weight loss and diabetes control? Somebody who has already had diabetes, again, we have seen people losing weight, 5-10% of their existing body weight. We know diabetes reversal programs, and one of the trials which have shown in terms of reversal of diabetes or diabetes remission is the direct trial. So as you see, the greater the weight loss, less than 5, 5 to 10, more than 15, the higher the remission of diabetes. So we are talking about cure of type 2 diabetes once you have the weight loss. But weight loss in terms of achieving this is always a problem. There are various methods. Again, orlistat is one of the drugs which is approved, but we know in our setting it may not be that helpful. Liraglutide, semaglutide, and naltrexone, phentermine, topiramate combination, all these are approved drugs. But we'll see the evidence with these drugs, how it translates in terms of our management aspect. So there are certain unmet needs in weight loss therapies. One is the sustainability of weight loss. We have seen patients losing weight and then regaining this weight in the long term. So a drug has to have a long-term sustainable result. In quality of weight loss, we need to have adipose tissue or visceral adipose tissue to lose, not the muscle mass and the other bone mass. Effect of concomitant anti-diabetic medications should also be taken into account. So when you're trying to address this unmet need, a molecule which can address this gap is again GLP-1 receptor agonist. How it addresses the ominous octet in multiple pathways we have seen, how it improves glycemic control and with the risk of minimal hypoglycemia, the cardiovascular benefits. Among the drugs, it's the GLP-1 receptor agonist which stands apart. And when you talk about oral semaglutide, there is a significant weight loss. Dr. Prasanna was talking about that. The mechanism by which is with the hypothalamic areas. The satiety centers are where the communication between the hypothalamic satiety and the appetite centers in terms of the molecular level has been now elucidated. So the trials have shown that semaglutide predominantly affects the fat mass. So if you see among the out of this 2.7 kilogram on either side, it's the fat mass which is lost. Out of that, the 2.6 kilogram is the fat which is lost, and that's what we want. The pioneer trials in terms of achieving the A1C goal, you have 3, 4, 7 and 14 milligram of semaglutide. The higher dose of semaglutide, 7 milligram, achieves the maximum glycemic control with maximum amount of weight loss. And this is sustained. You see the results up to 36 weeks and 26 weeks and 52 weeks. So more than a year, you have sustained weight loss, which is again significant. I think Dr. Prasanna touched upon this slide where the change in body weight is compared to other drugs, it is remaining. And over time, even these are trials which have got more than a year, 1.5 years, 52 weeks of trial results, we have had significant weight loss. Even on top of the existing drugs, even on patients on any of the other anti-diabetic medications, be it empagliflozin compared to sitagliptin, compared to liraglutide 1.8 milligram, the blue bar actually falls down the maximum. You have got maximum weight loss with oral semaglutide 14 milligram. Even on top of the background medications, so regardless of whatever oral drugs they are on, regardless of whether they are on insulin or not, semaglutide achieved significant weight loss. So the change in HbA1c, again we are talking about anti-diabetic medication, the change in HbA1c is also preserved compared to placebo, compared to liraglutide, oral semaglutide, again the 14 milligram dose achieves significant reduction. So this has made the standard of care. We know the cardiovascular risk reduction, and the weight part is also mentioned in this. So the aim is to promote weight loss and have minimal weight gain. It's the GLP-1 receptor agonist, which is the preferred agent. So let's end with the patient profile. A patient named Pavan, who is an average youngster, a relatively young patient coming to our OPD. 46-year-old software engineer, so obviously we are talking about sedentary job and snacking and other things. He is already obese. The BMI is 30, and he kinds of doesn't follow the lifestyle as advised. He's got a strong family history of cardiovascular disease also. So he was initiated as per the standard of care on metformin, diet and exercise. But what has happened is that he has progressed. We'll see that we allow him to progress, and 3 years later he has come back with this. Again, if you see his lifestyle modification is rather poor. 2 to 3 times a week for 20 minutes. This is hardly the exercise that we advise. And look at the treatment regime. He has gone up with the glimepiride 4 milligram once daily. So metformin plus glimepiride. So this was what was practiced probably 3 or 4 years before. And over the past 3 years, he has had weight gain and hypoglycemic episodes on the background of his cardiovascular disease. So this is what happened after 3 years. So from 88 kilos, he has made 91 kilos. His HbA1c is remaining uncontrolled. So we have made a treatment decision at this time where his A1C has grown over years of, and then he was added a GLP-1 receptor agonist and statin was added. So yes, it has yielded result. His HbA1c is coming down. It has reached the target of less than 7 over years. But what has happened is that he has already had the risk factors which have driven him downstream, as the pathway shows. So this is after 3 years of adding a GLP-1 receptor agonist. He has achieved weight loss, 91, he has got 78 kilos. His targets have improved, he doesn't have albuminuria. But see, what has happened is that he has already developed cardiovascular complications. At that time you are intervening, and probably we are doing good. At least at that time we are intervening, and we know GLP-1 receptor agonist and SGLT2 inhibitors actually prevent further complications. But already he has had a cardiovascular complication. But what would have happened if appropriate measures, like what we are talking about, the upstream pathway, if we had added effective drugs which would had helped him to lose weight, like oral semaglutide, we know how effective it is. So apart from metformin, diet and exercise, if oral semaglutide and statins was added at that time, it would have had prevented his cardiovascular disease onset and had better result in terms of preventing further complications. So this is the upstream approach that I think we should start doing now, where we are not allowing the patient to develop complications or increase in the risk factors, as the guidelines have suggested, high risk for ASCVD and high risk or already had an event. We shouldn't wait for that. We shouldn't wait for an obese patient to develop that, and we should allow him or give him the benefit of GLP-1 receptor agonist. Now that oral semaglutide is there, it works well in these kind of settings. So I would end here by summarizing that the complication, the twin epidemic of diabetes we are facing, and managing high body weight right from the onset, right from the pre-diabetes state itself is important. And it's an upstream approach that we should try to follow. We should try to follow this and try to prevent these patients from going into, in terms of the higher cardiovascular risk and cardiovascular event probability. And oral semaglutide has made a path-breaking change in terms of that, where we help the patient to achieve this weight loss with an improved cardiovascular risk profile and safety profile. Thank you.
Thank you, Dr. Jaiprakash. Anyway, it was a wonderful talk. Anyway, semaglutide is a very promising drug. I think in future it will be definitely a good hormone for the diabetic treatment.
Thank you, Doctor, for this excellent presentation. So, running short of time. Delegates, any questions? You can please ask the speaker. If there are no much questions, we'll end this session now. Thanks for this.
Thank you, speakers and chairpersons. So, So next talk will be by our dynamic speaker, Dr. K. N. Manohar, who is going to speak on decoding and preventing CV events in dyslipidemia management. So for this, I would like to invite chairperson as Dr. Subrata Das.
Well, I'm the last man standing. Dr. Subrata Das, you want to make a comment before I start?
Dr. Manohar, first of all, congratulations to you for wonderful organizing a scientific program for us today. We are eager to hear you. Over to you.
Thank you. Well, cardiovascular disease, as we all know, is the major cause of mortality worldwide. Some points I would like to make. We traditionally do a fasting lipid profile, but even a non-fasting lipid profile has equal or even more importance. Even the Lipid Association of India guidelines says, if you do once the fasting, on the follow-up, you should do the non-fasting. Because most of the time we are in the fed state. Especially we start having a late dinners, we are almost always in the fed state. So non-fasting has a role. And a lot of data is coming in that non-fasting could be better reflector of mortality. I wanted to keep it short, but not this short anyway. Non-HDL cholesterol is emerging as an important entity. So I always put these points to ponder, whether we really look at fasting and non-fasting. And most of the time, dyslipidemia management is based on ASCVD risk reduction. We have moved away from just looking at the lipid. Rather, we are looking at ASCVD risk reduction. But if you look at the guidelines, it all is based on calculators. So do we really use the calculator? But actually, there is only one indication where you need to use calculator when is a younger patient and a non-diabetic. If he is a diabetic, almost always we give the statins. And if someone has a secondary prevention, no question, statin are the number one drugs to be used. So there is always a minor difference only two in the triglycerides, where you can see 26 milligram per deciliter difference versus fasting to non-fasting. It also depends on what food we ate and how long after the food it was measured. And interestingly, the fluid intake also determines the HDL triglyceride level. Otherwise, if you look at the total LDL and non-HDL cholesterol, it will be showing slightly lesser than in the non-fasting state. Dyslipidemia, we all know, is a major concern, and very important point I would like to highlight is very high risk group has been formed now, and they are looking at targeting LDL of 50. Or someone had a coronary event, secondary prevention, and is a high risk or a second event, then you need to look at LDL of 50. So iceberg is always shown in all diseases. We are always treating the tip of the iceberg, and we are not looking at the base. And definitely, even though we have seen good LDL levels, means less than 100, sometimes still people have ASCVD. So what is the residual risk? Even after statin treatment, if you look at the left of the screen, 77% unaddressed residual risk is always there. So coming to the molecule per se, bempedoic acid, adding bempedoic acid to statin therapy, or using the bempedoic acid in statin intolerant patients. The first group is what we need to firstly look at is people, if you look at the bottom half of the screen, Clear Serenity and Clear Tranquility are the two trials which used bempedoic acid in people who are intolerant to statin or who didn't tolerate maximal doses of statin. The top half, Clear Harmony and Clear Wisdom, are the two trials which were used independent of the statin therapy. So what is the difference between this Clear Harmony and Clear Wisdom? Clear Harmony was safety-oriented trial to look at the side effects. Clear Wisdom was efficacy-oriented trial. And in both, you can see around 15 to 16% LDL reductions were seen. So percentage change from baseline to week 12 in LDL, because we are looking at the efficacy, is about 15 to 25% in different group of people, is the efficacy of bempedoic acid with respect to LDL therapy, LDL reduction. So in all, what they showed is clinical trials have shown that bempedoic acid monotherapy or in addition to background lipid lowering therapy lowers the LDL cholesterol, non-HDL cholesterol, and it has some effect on the ApoB and HSCRP, to an extent slightly better than what statin does. So where does it act? It acts, simple way to remember is we all know statins are HMG-CoA reductase inhibitor. 2 steps above that is what bempedoic acid acts. One important highlight what we need to look at it, I don't know if the pointer is working, it won't not, is onto the left of the screen here, AMPK activation. We all know statin associated diabetes or NOD, new onset diabetes in statin therapy is a problem. Where bempedoic acid scores over statin definitely is, it possibly has a better effect on the glucose by AMPK activation, where metformin also possibly acts, and it has a favorable effect on reduction of the glucose as it's shown on the top of the left extreme of the screen, reduction of glucose. That is one point which we need to remember. So again, clear serenity and clear tranquility are the 2 trials. They are both efficacy trials. Serenity trial was against placebo. Tranquility trial had an arm with bempedoic acid plus ezetimibe. The other arm was placebo plus ezetimibe. So we are trying to look at the efficacy individually of bempedoic acid and with ezetimibe how it does. If you look at the number of patients in this trial study group was much less, but it is heartening that it reduced the LDL cholesterol by 23 units in both studies. So clear serenity was versus placebo, tranquility versus plus ezetimibe plus minus the drug. So if you want to look at the overview of bempedoic acid, harmony and wisdom and serenity and tranquility are the 4 trials which are there. And heartening to know it has been approved by most of the regulatory bodies, US FDA, European Association, and it comes in 180 milligram of tablet and it can be taken once daily irrespective of the meal. The much awaited clear cardiovascular outcome trial, it is the CV risk reduction trial. We know CV outcome trials. Statin is one of the important drugs to reduce the CV outcomes. Bempedoic acid, very, very recently, few weeks back or few days back, this trial results were out and it reduced the 4-point MACE. So that is one big highlight about this molecule, that in the future it could be very, very useful in people who are intolerant to statin or who are having a load, not tolerating the full dose, then you can add on. Or I feel another area is IGT, people who are on IGT, and if this has shown some benefit with respect to mortality reduction, instead of using statin, can you use bempedoic acid? That is the reckoning question what comes. But of course, there is a huge amount of data with respect to, I'll skip this slide, amount of data with statins. But there is some controversy. Statins were always controversial molecules, even though we have used it for decades. That is because of the 28% increase in the risk of developing diabetes with statin therapy. That was shown in the Jupiter trial. So that is where bempedoic acid scores over the statin, that it is glycemic friendly, helps reduction in LDL, and most importantly, the clear trial has shown that it reduces the CV outcomes also. The 4-point MACE reduction is significant. Thank you.
Thank you, Dr. Manohar, for a wonderful talk. So I think this will be very useful to us in patients who have diabetes, particularly with statins causing some amount of hyperglycemia. And many people don't tolerate, say, rosuvastatin more than 20 mg in among Indian population. LDL more than 100, that forms an important thing.
Yeah, rightly so. That residual risk reduction still, if it is looking at, this could be a good add-on therapy. And then, of course, if you are looking at IGT, people treating for dyslipidemia, with the CV outcome trial in a positive trend, I think we can think of using it.
Another thing will be to look at how it is helpful in our non-alcoholic fatty liver diseases.
I don't think we have any data as of now on NASH. We'll think
Who
about
knows?
that.
We can always do some study to find out.
Sure. Sir, can you get the mic, please? Or you can come forward, sir.
Sir, some of our patients are showing a lot of high triglyceride levels. Although LDL is normal, HDL is normal, triglyceride will be high. What about this drug on triglyceride level?
A very good question. Triglycerides in itself treatment, actually we over-treat it. LDL cholesterol is the first thing to look at it. If it is coming into the high risk range, more than 100 now is the guidelines, or if there is no other major risk factor, more than 160, definitely treat it. Triglycerides alone, if you look at the indications for treatment, if it is more than 500, we need to treat, more to reduce pancreatitis. Between 250 to 500, if the HDL level is very low, there is some benefit. Again, it could have been a debate topic on how triglycerides are useful. If it is 250 to 500 and HDL is very low, then there is an indication for triglyceride treatment. Otherwise, triglyceride treatment has not shown to improve mortality. Yeah, that's what the new trials have shown. If the rosuvastatin is stronger. Yeah, that's why we need to monitor it and titrate it accordingly. If that is the case, previously we used to stop it. Now we can think of adding or substituting bempedoic acid in such cases.
Actually, even in patients with fatty liver disease who are on rosuvastatin, you give them vitamin E and acetylcysteine, they actually do better. And sometimes if you're worried about triglycerides, low HDL, saroglitazar has shown some useful.
Yeah.
And what about this established atherosclerotic cardiovascular disease, or probably patient coming with a acute coronary syndrome or post-MI for stabilization of the plaque, despite the LDL may be normal or maybe only...
No two things, statin are the number one drugs. So please don't go away.
No, somebody is not tolerating the high dose
not
of statin.
Not tolerating, yes, definitely
so
we can.
There's a role of the plaque stabilization in those
Actually,
conditions.
the Clear Harmony trial was used with a lower doses. Some people couldn't tolerate, the lower dose was rosuvastatin 5 milligram, only they could tolerate. On top of that, adding bempedoic acid showed a significant reduction in the LDL level.
So probably low dose of statin and then this,
bempedoic acid. Or if someone is not tolerant at all, I don't think so many cases we see. I have used only one prescription till now, and Prasanna sir said wait for at least 5-6 prescriptions to know what it
So
does.
probably cardiologists are going to give the input for that, acute coronary syndrome cases. Yeah.
All right.
Perfect.
Yeah, this is a small trial which has shown the CV outcome results. Statins have been there for decades together, and one of the number one drugs to stabilize plaque and reduce the secondary prevention after aspirin is statin. So with the wealth of evidence, we should not just deviate from it. Let it prove its worth. Again, we have said it started all with people who are intolerant to statin or not tolerating the maximum dose which you recommend. So that is where it is. It is not the number one drug to be used, definitely no.
Okay, there are one or two odd things. If I don't know how many of you use the ACVD estimator, but if you see that it's always from 40 to 75. So if somebody is less than that, you don't know what to do. If you have a risk factor like a diabetes and all, there is no question about it.
True.
So, but otherwise there's a hitch there, number one. Number two, just to explain, the potency of each statin doubles. Lovastatin is considered as 1, then simvastatin is 2, then atorvastatin is 4, then rovastatin is 8. So it's almost doubling. Second thing is that the metabolism of rosuvastatin is different from atorvastatin. So if you find that the enzymes go up after using atorvastatin, you can switch to rosuvastatin. Am I clear?
The lower potent statins have lesser side effect on the liver, in short, if you want to get what sir is saying.
That is the important thing you must know. And there is the question of having myopathy is a myth. It is not true. If you read any Harrison, Cecil, any book, you will find that there is something called a nocebo, where they say you tell them there's nothing going to happen, you will have, patients will have no problem. But if you tell the patients, look for hepatitis, look for myopathy, look for enzymes, check the CPK, they will come back with you. This is the experience I have had. So you don't sell them anything, look for cataract and say you may develop diabetes. But be careful, when you say you may develop, that's a deterrent. Okay? See what I...
If you are in the target, you don't have to switch over the statin therapy at all. It is same as for atorvastatin. I don't think there is a different indication.
See, the book again says that you can use up to 40 milligrams of simvastatin, that you should not use. You can use up to 40 milligrams or 80 milligrams of atorvastatin. But when you start going to higher doses, it's better to switch over to rosuvastatin because the metabolic pathway is different. Then switch to see, this myopathy is a class effect. It is not a single drug. Okay, so 95% of the time it's a nocebo. You tell them nothing will happen, nothing will happen. This is, I'm not quoting you read Harrison, you read Cecil, it's written there. Then you switch to rosuvastatin and see. You can use ezetimibe. You can use ezetimibe, which is not so potent. This is too new. It's very new, so we don't. There's a saying which I have always quoted: when a new drug comes, wait, don't. I will use it for everybody. Then after 5 years, effects will come. I will not touch it. It could be now Japan, Korea, US, Europe. Please remember, US FDA is the strictest of all. After the thalidomide tragedy, the US FDA is the most strict thing. Now the European Medicine Agency, EMA, is equally strict now, but still the FDA is the strictest.
Over to him.
So I'm not going to leave Dr. Manohar so easily. So he keeps troubling all the speakers with questions. So
I don't think so.
So some comments, that's all.
Sure.
Uric acid, you didn't touch the uric acid. Bempedoic acid, only known problem is with uric acid. Tip of the iceberg, we keep talking about the tip of the iceberg. Any tip in India is bigger than Madagascar. So we should remember. So there is no tip in India. So how much non-fasting is you are going to check the lipids? Say 1 hour after the meal. What is the quality of the meal? Because for diabetes, we say 2 hours after whatever is a balanced meal, we check it. So if you are going to check non-fasting state, so
it is irrespective, there is no time limitation for that. What Lipid Association of India says is, once you do fasting, next time you do random.
It's a random.
I think the general advice is, don't modify or do something to your diet because you're going to do the test. Okay, do what you have been doing regularly.
After regular meal.
Regular meal.
And ASCVD is not relevant. We cannot say it's not relevant because we have, particularly being in Bangalore, every second patient comes with a big health checkup brochure. So they don't have any other problems. They come with only a abnormal lipid profile. So those patients, it helps us make a decision. So ASCVD risk scoring is very important for those patients. And good session to end. Wisdom, harmony, serenity and tranquility. So I think
I think it sums up the day one of KRSSDI. I'm very, very thankful to everyone. Late in the day, and we started quite early, and then most of the sessions were jam-packed. It can't happen without the delegates being present. I can see a few of my students, but I can promise that I have not threatened them that they have to be there, but they are there. Thank you so much. Thanks for the chairpersons and everyone for making it grand success. I would invite everyone for a unique mind and body workshop. I've been telling it throughout. People must be bored by listening to me. 7:30, 7:45, be here and experience the difference. And tomorrow also will be interesting, and I invite everyone to come back and join us tomorrow. And we will have AGM in 15 minutes downstairs.
Thank you. Thank you, Doctor.